That's a very curious show of hands. How many of you have completed the mock paper? Okay, okay. You'll give me a very surprise look. I give you an even more surprise look. Good job. Yeah. You're like, you mean we were not supposed to? No, no, you were supposed to finish a mock paper. But it's fine if you don't. Wow, I've 18 online participants. Hi, everyone online. Nice to meet all of you. If you have any questions later, please, my advice is to unmute yourself and to just share it. I have the chat box open, but it's a bit hard to see later when I teach on screen. So yeah, just unmute yourself and ask me a question later. Hi, welcome. Your name is? Which school? Oh, VN, NYJC. You happen to know them? Nice to meet you. VN, if you want to see. 4 more minutes. No, 1 more minute. Okay, hi everyone online. Yes, this recorded video will be uploaded onto MeWatch, available for you to watch when you're free. No, just kidding, not superstar yet. But yes, it will be available online. So if you're at home, you can listen. If you happen to be multitasking while you're at home, you can check back later and watch. Yes, but thank you for the question, Evelyn. If not, I am going to start. The first thing I will go through today will be the mock paper. All right. Okay. Okay, if not, welcome everyone. Let's wait for Kanishka. No pressure, no pressure. Hey, don't stress off that. It's a holiday, man. It's a school holiday. First thing first, I hope all of you wished your secondary school teacher happy Teachers Day last Friday. If you forgot about them, just kidding. Okay, let's start. Before that, yeah, hi, welcome to Overmark. For those of you who are here for the first time, I'm not in the picture, but she is. Yes, she's there. So she's here for a second time. And that's our ops person. Hi, Marcus. So we teach secondary school, IP, and JC tuition. That's me. I believe for those who came here the first time, the photo has been upgraded. I now look much better. Previously, I looked like a train wreck. Okay, a bit about me. I teach JC Bio. I'm a former MOE teacher. I was her teacher. So yeah. So you that old? Yes, I'm that old. Okay, so I've taught more than 300 students for the last 9 years in bio project work. And quite a few of my students do well. The only fun fact you have to know about me today, which is linked to today's lesson, is I like her and I like her. You know who they are? Who? Yes, this is Bay Suzy, not my Bay lah, but her name is called Suzy. And that's IU, yes. I did not manage to go to her concert because your generation stepped up all the tickets and I was very sad at home. But oh well, there's free songs on YouTube. Okay, so you're thinking, why do you all need to know about these two ladies later? You will see why, especially during respiration. Okay, we have a lot of students. We have practiced all over Singapore. We have 7 now. Tampines is up this week, same as Jurong East. So my dear JPJC folks, there is some hope. Okay, all right. Next, yeah, this is us. I'm gonna skip this. And these are our resources. I think some of you have already tried the free notes in O levels. The notes you have today are also free. So we have a lot of different resources for students who are not with us, as well as for students who are with us. Okay, and with that, I am going to shift straight to the lesson for today. All right, so can I get you to take out your mock paper, please? For that, let me off the music. Okay, for those who have done it, you can self-mark your answers first before I go through. I will be going through everything, so don't worry about that. Yeah. One, I put the chat there. All this for them to pray to the infant. Actually, he handled giving out some people something that. Okay, you're done. Anyone needs more time? Yeah, I see Yan still trying to copy something down. If not, I shall begin. So first thing first, Satya, what's the first thing you should do when you do MCQs? I'm asking you because you're the first bro. You're the only bro here, bro. So today we're going to have a lot of discussion. So what's the first thing you should do when you do MCQs? Thank you very much. Next, after you read the question, what do you do? Pray really hard to study it? No. Okay, so I'm going to introduce to you all today, when it comes to MCQ, especially at A-levels, it's not so much about finding the right answer. Some of the answers are actually, so rather some of the questions are actually meant for you to find what is the least wrong. And there's one example here later. So that is the biggest difference between O-level and A-level biology. What I'll go through later is a technique where you basically have to find out what is wrong with every option. And if you find 3 options where things are wrong, you are probably left with one that's correct. Okay, so let's start. Now, question 1. This one is a very nice warm-up question over here. It tests you on microscopy as well as cell ultrastructure. Now, they have told you over here that chloroplast is about 2.2 micrometers long. Okay, now, then they're asking you, okay, sorry, the starch grain here is 2.2 mm. When I first saw this question, I thought, okay, actually it's not very hard. Why? If this thing is 2.2 micrometer, what is the only available answer that's possible over here? Okay, now can it be 48 mm? Not really. Why? If I use scale here, if this line is 2.2 micrometer, okay, and I put it here, it's roughly about here. Okay, and this is roughly about here. The closest answer here will be 4.9. So the proper way to do this would be to take your ruler, measure this starch grain, find out how long it is on your ruler, find the length of T and U, okay, and measure it and do the math. But this answer actually is quite straightforward. 48 mm is 4.8 cm. You all know how long 4.8 cm is? Can your chloroplast be 4.8 cm? No. So everything here is gone. Okay, then 16.5 cm is way too long. Okay, so the answer here is B. Clear? Bigger. Now, question 2 over here. It's a question where they're asking you to find similarities, okay, between chloroplast, mitochondria, and a typical prokaryote. When I see these 3 things, what is 2 things that should come to mind? Firstly, ribosomes. Okay, what is so special about the ribosomes in a chloroplast, mitochondria, and a typical prokaryote? Amelia. I'll give it a try. What's so special about the ribosomes in a chloroplast, mitochondria, and a prokaryote? Okay, so prokaryotes have 2 sizes. One is 80S, one is 70S. What is the ribosome size for these 3 things? The ground? Okay, yes, 70S. So the easy way to remember this is very straightforward. Bacteria is smaller than us, so bacteria should have the smaller ribosome, 70S. So all eukaryotes, this is in eukaryotes. And why do I just focus on this? Once you know ATS is only in eukaryotes and not in these three things, the question becomes so much more manageable. Why? If I look at the options now, three options here have ATS. Immediately, they are out. But let's confirm a few things. Now, if I put C as the answer, once a prokaryote, it has circular DNA, mitochondria has circular DNA. True or false? True, right? Okay, this is true. Mitochondria, circular DNA, chloroplast, this is also true. That's why C is the answer. Okay, so like what I said, it's not an issue of knowing what's correct. Just find out what is wrong. And once you know ATS is confirmed wrong, answer is C. Okay, let's go to question 3. Over here, okay, the answer is D. For typical questions with tables, especially with bonds, okay, don't keep the thinking in your head. What do I mean? Most students like to think, what did I learn for this? And they try and think and think. I was a weak student in school. Hi, welcome. Just take a seat, no worries. You started first. Your name is? Yeah. Wow, that's hard to spell. How do you spell that? Hiranya. Thank you so much. What an interesting name. Okay, you are friends. No. Now you are friends. Okay. So why do I say this? I was a very weak student in JC. Okay, why? Circumstances were different, but don't worry, I'm a pretty good bio teacher now. So I've learned as a weak student, what I kept doing wrongly was keeping all the information in my head when I was doing a question. So when I can outlook everything, first thing I do is this. It's a question about bonds. Let me write down where do I find an ester bond. Now in your syllabus, only one thing is an ester bond, lipids. Hydrogen bond, this is a bit tricky. We know we find it in proteins. Needy. At which level of proteins do I have hydrogen bonds? Secondary structure, very good. Okay. Next, let me ask Jiaying, where do you find hydrogen bonds for cellulose? Sure, you mean hydrogen bonds for cellulose? I did not know that. Yes, there is. Where? Okay. Come, anyone? Where do I find hydrogen bonds in cellulose? Okay, you find it right over here. So if this is my this is my cellulose chains where they are straight, there is interchain hydrogen bonds between them that keeps them strong. So this is the difficult part about this question, because most students will have forgotten about that. You also find these hydrogen bonds, okay? Yeah, okay. So cellulose has these hydrogen bonds to keep them strong. And that's why when you eat a vegetable, it feels crunchy. Okay. Next, disulfide bond only in proteins. Let me ask Raylene, at which level of a protein structure do I find disulfide bonds? Good. Another one? Is there another one? Quaternary, thank you very much. Okay, on that note, can I check with you guys, how do you spell quaternary structure? How many R's are there? Very easy, I'll ask you this. Ding, ding. Difficult question in promos. After the A, is there an R or not? Okay. Please confer with your friends. Is there an R after QUA? Okay. How many say yes? Sit down. That's it. Okay, the answer is no. There's no R here. And what the easy way to do this is very straightforward. I teach with a lot of humor because why? If I just choose a life example, I don't think you can remember anything your teacher taught you in primary school, but I'm sure you can remember a very stupid and silly incident. I can. So I believe in teaching with humor and doing stupid and doing silly stuff because you will remember it. So how do I help you remember this? How to pronounce this level of structure is called Qua. Okay, phonetically, how do you spell Qua? Qua has no R. Okay, so QUATER, quaternary. If you don't believe me, I have a current student called Lucas. He's a very muscular, macho guy. Okay, biceps very big. He follows this. And every time in class, when I ask him, quaternary got R after that, he will go. And he remembers there's no R. Okay, so there's no R after quaternary. So now, once you know, easy, lipids only got ester bond, A and B straight away out. I'm left with just C. Okay, then amylose. Does amylose have hydrogen bonds? Amylose only has glycosidic bonds. Okay, all right. The answer here is D. The answer here is D. Okay, and that's why this is out. People online, okay? It's not for camera, sorry. Next question 4. This is a very, very complex question because it plays with the mind. Now, they told you firstly that they have 3 different organisms adapted for different climates as shown below. I have 1, 2, 3. Like what I said earlier, make your thinking visible by writing down what usually happens to the membrane. So, Hiriana, sorry, I massacred your name. Okay. In a very cold climate, is the membrane too fluid or too rigid? Good. How to remember that? Very simple. When you're very cold, right now, very soon, in about 20 minutes, right? Then I go through the question, because it's so cold and comfortable, do you feel like moving? No, you just feel like sitting there, not doing anything and falling asleep. But if I turn on the aircon, very soon you will do this. Very hot. You go to tie the hair up because the neck very hot. Okay, I know because I got a man bun. So if the analogy you can remember, when it's very, very cold, just like you, membranes become very rigid. When it's very hot, just like you, membranes move a lot. So first thing first, what I will do at the site will be to write down very clearly, because I have a poor memory, that the membrane is loosely packed when it's colder. Okay, all right. All right, I need to be loosely packed. I say again, I need this. I need this. Because the membrane is too cold, it's too rigid, I need to loosely pack it now. Next, as what I mentioned just now, when it's too hot, it is too fluid, I need to tightly pack it when it's warm. So whatever I'm writing down here at the side is not the condition of the membrane. It is what the organism needs to do to the membrane. Okay. So if I know that I need it to be very loosely packed when it's colder, now if I compare between 2 and 3, this organism has very loose membranes. Which one do you think is colder, 2 or 3? Let's ask Gabriella. Don't worry, I guarantee you by the end of 3 hours, I will have caught you at least once, maybe 2 times. Between 2 and 3, which membrane is less tightly packed, Gabriella? 2. So would that be a colder climate or a not so cold climate? Good. So here will be the coldest. Then 3 will be colder lah. All right, then now comes to 1. 1 is just normal. Okay, it's normal. Normal also means it's a bit more rigid. So if it needs to be rigid, it's probably going to be in a very warm climate. So with that in mind, we can now go to the questions and we see which particular category shows where 2 is the coldest or 2 is colder than 3. Okay, the answer for cold, 2 must be cold. Straight away, desert's out. Okay, 1 must be warm. Okay, Arctic is out. Now then between 2 and 3, my 2 must be colder than 3. Okay, therefore temperate and Arctic. For those of you who didn't take pure geography, Arctic is -40 lah. Temperate is Switzerland, Europe, very nice. Okay, all your K-drama when they wear the trench coat, right? That is temperate country. Okay, so therefore this order is lopsided, it's wrong. 3 can't be cold, no, can't be hot, so it's out. That gives you answer. Okay. 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 Now if you're wondering, Mr. Paul, your handwriting is a tad challenging to understand. It was a difficult choice between choosing to be a teacher or a doctor lah. But life chose for me, I became a teacher. Okay. Now, but don't worry, it will get better. It will get better. Now, for this question, I call it a question of spotting the lies. You know, sometimes in life, when your friend tells you a story, like, I'm sure after September holiday, you go to school on Monday, you ask your friend, hey, what do you do in the September holiday? Oh, no, I didn't study lah. I went to JB for holiday, spent time with my family. You're like, liar, confirm closet mugger. Okay, all the WA you say don't study, but you score super well. So this is what this type of question is, spotting the lies, or rather spotting what is not written there. Okay, so they tell you over here, graph shows the temperature of helical structure of collagen without hydroxyproline. If you don't know, hydroxyproline is one of the 3 amino acids in a collagen structure. Coming soon, Leting, what is the other 2 amino acids? Okay. Just remember, collagen very small, needs to be very tight. So what's the smallest amino acid? Good job, glycine. Okay. Coincidentally, glycine also. Okay, so it's glycine. The other one, okay, just proline. Okay. So over here, without hydroxyproline and without hydroxyproline. Now, this is my next tip for all of you. A lot of you spend a lot of money, especially the ladies, on highlighter. Gentlemen got one highlighter in school, very good already. Correct, Satya? Hey, how I know? All my boys in school last time they have highlighter. I have to buy for them. So since you have so much highlighter with different colors, it's time to use it. If you see, got 2 graphs, dotted line, highlight the dotted line in one color. That is my first pro tip for today. Very simple primary school tip, but it saves you a lot of heartache later. So that you know exactly what in the world am I looking at. Okay? Now, people don't do this, increase their chance of spotting things wrongly. Okay, so now they tell me, which statements explain the importance of hydroxyproline in the structure of collagen? Now we're just looking at the facts. Presence of hydroxyproline increases thermal stability. How do I know what is thermal stability? Here. As long as it's up here, this is thermal stability. The moment it decreases, or my percentage helical structure decreases, in short, this is denaturing. So 3-0 here means everything is denatured. Right? So you can see clearly, blue color one with hydroxyproline. Up your temperature, apologies. Up your temperature of near 40 degrees, it is still not denatured. But for the collagen without hydroxyproline, at about 20 degrees, you are kind of in trouble. So, sorry, where do you find collagen in the body? Skin, right? Okay. So, can you imagine if you have collagen without hydroxyproline? My aircon temperature now is 18 degrees. So, 70% of your body will now be on the floor because it will have melted. So, first, yes, it increases thermal stability. 2, at higher temperatures, collagen with hydroxyproline results in less silices present than without hydroxyproline. Okay, at higher temperatures, basically it's over here. 2 ways to infer this. I have no idea what is higher temperature, but I'll assume very, very high over here. Okay. Yeah. Okay. They're saying this one here at higher temperatures, they have less silices than those without. Is that true? No, right? It should be the other way. Okay. I went a step deeper. I thought very deeply. I was thinking maybe the counting number of helices. Can I count the number of helices here? So cannot. But either way, this is wrong. So 2 is out. With 2 out, we can look at the options. Okay. 2 out, 2 out, 2 out. Life is brilliant. But please check option 3 to make sure it's correct. Hydroxyproline contributes to extensive hydrogen bonds that hold the helices together. In all honesty, the question never say anything. But if you know 2 is wrong, okay, like outright wrong, okay, and 3 is plausible, like, okay, the reason because I have better thermal stability could be due to the hydrogen bonds. I put this as plausible. Then now it's choosing versus 1, 3, which is 1 is confirmed correct, and 1 is plausible, versus 1 and 2, which is this one is correct, this one outright wrong, right? You choose the more plausible one. Okay, like I said, A level is like that. Okay. Now, question 6, this one, okay, for context, this medicine is meant to treat alcoholism, if I'm not wrong. So what's the best way to make an alcoholic person stop drinking alcohol? If you read the whole story, at the end of the day, they say, oh, if you eat this, alcohol cannot be broken down, right? You get nausea and you vomit. So in short, this medicine makes you puke. So the more you eat it, the more you drink, the more you puke. So if you keep puking, do you like that? You don't like that, right? So you start drinking. That's what the whole story is about. Okay, focus on the purple parts. So the whole story is very long, but in short, this disulfiram, okay, has a Michaelis constant for acetaldehyde dehydrogenase. It increases this. Okay, this, once you see Michaelis constant, is just affinity. Okay, right now, also, because they tell you that this whole entire reaction is catalyzed by the enzyme, is very specific. If it's very specific, okay, why is it important that the enzyme emitter is specific? Now, if it's specific, according to the blue portion and the purple portion, it only breaks down one reaction. So if it breaks down only one reaction, the story, this is correct. It cannot disrupt other metabolic pathways. Okay. Now, 2, it prevents alcohol from binding to the active site. Nah, this is outright wrong. You all know that this is the substrate. Alcohol is not going to bind there. So this is wrong. Okay. 3, it is unlikely to cause unwanted side effects. Now, this red color part is a side effect that they want. So when you were doing this, you were thinking, sure, isn't that a side effect? There's a side effect there, but so why is 3 correct? No, they want this side effect. Now, what I told you, this is to stop someone from drinking. So I'm making you puke when you drink. So every time you puke when you drink, you won't puke anymore because you don't drink anymore. So this is not an unwanted side effect. Okay, so this is correct. They only cause this side effect. Okay, then 4, it binds to the allosteric site of this enzyme. Once again, the question doesn't say anything. Okay, I cannot infer that from the question. Therefore, it's out. 1 and 3 is the answer. Okay. Now, question 7, this is very straightforward. It's about stem cells. Just take note. If 3 is a subset of 2, it means 3 can do everything that 2 can. So it means that 1 has the largest potential. Which stem cell has the highest potential? Zhimin. Yes. Which stem cell has the largest potential? No. What's the name of the stem cell? Wow, Mr. Paul so heng. Let me ask Sing. Sing. You are Sing. Oh, Sing. Sorry. Which stem cell has the most potency? Or has the best potency? Totipotent, very good. Okay, so sorry, my JP folks, you never learned this. Okay, so it is totipotent. This must be totipotent. Now, so once you know this, okay, what is the ability of a totipotent stem cell? It can form the whole organism. Okay, so once you know it can form the whole organism, this one is out. Okay, this one is out. Then next, what's after totipotent? Kanishka. Pluripotent. So what's the definition of pluripotent? You can differentiate to any cell type except the placenta. Okay, so almost any cell type. But here, so this one is correct. Okay. I look at A. Differentiate into a limited range. Straight away wrong. So everything is gone left C. Okay. Then for 3, differentiate into a limited range of cells and a tissue appropriate to their location. This is your typical multipotent stem cell. Question 8, wow, why am I so colorful? Yours not so colorful. This is what I will advise you all to do. Same thing with your highlighter and color. If I have a question like this, where there are 4 different drugs and I have 4 different tables, I will match it together. So decrease pool of available nucleotides that inhibit the chain elongation. Which drug does that? Okay. If I want to increase my length, I need nucleotides. This one prevents production of nucleotides. This should be together. Green, DNA strands not available for templates. Means what? Still together. Prevents unwinding. DNA damage during replication and cell death occurs. Damage, repair. Red are together. Blue, exposed DNA template strands are unable to be copied. If cannot copy, okay, 2 parts lah. Maybe can unwind, can separate. But my DNA polymerase cannot do anything about it because it's being inhibited, link there. Once you match the colors together, there is only one available option. So use colors to help you for such questions. It becomes more manageable. Okay, people online, I hope everything's all right. Any questions, just let me know. The people here are dying to ask me questions. Okay, same thing, question 9 over here. This is a very, very cheap question. When I first saw it, I was like, wow, the school that did this are big brain, big brain. Can I ask who got this correct? Oh, nobody. Okay, nobody. Now, very simple. First thing first, I need to show you, so you mean there are two parts where the DNA and where there will be bond break between complementary bases? Now, for option number 1, okay, now, when do I have the first break in complementary bases? When I break the bonds between my DNA double helix. First one. So then what's the second one? Second one is after your RNA has very beautifully formed, before it can be released, you must break that bond again. So there are two bond breaking there. Then now you can escape. So that's the first part where there are two bond breakage of complementary bases. Next, okay, bond forming is the reverse. Now, when a different color, purple, when my RNA nucleotides, okay, find the complementary bases, there is formation. First one. Secondly, after this RNA has discovered that it's done and disappeared, will this helix remain separated? No, there is bond formation here again. So that is the second bond formation. Okay. Now for sugar phosphate bond forming, okay, there isn't a double number of times. There's only once. When you form your RNA, your mRNA once, this only occurs one time. Phi nucleotide pairs with complementary nucleotides also only appears once when the formation of your mRNA. That's why the answer here is A. Okay? So copy down this thing aside. That's the helpful part. And left, Italy, weird. Okay, so far? Yeah, done. Yeah, I'm waiting for Gabriela. She's copying very frantically. No worries. Okay. Question 10. Okay, control of gene expression. None of reassure you as much as you all think control of gene expression is the nastiest thing this year in bio, it's actually very approachable. It's quite straightforward. Okay, later I'll teach you all. But in short here, they are telling you a lot of things. It's quite a lot of examples, but they're just telling you, oh, if I got a plus sign, it means it transcribes faster. So over here, these are 4 true statements. If I only got P and have a plus, what does it mean? Plus means there is transcription, basically. Plus means there's transcription. Then everything here has 4, 4, 4. In short, there are a lot of things that's increasing transcription. Okay, so now they're asking you to make a valid conclusion. Firstly, they ask you, okay, right? So E is your enhancer. They have mentioned that an enhancer is required for transcription. Which option here, okay, shows you that transcription is possible without an enhancer? A, right? Yeah, no enhancer. Straight away, gone. Rate of transcription is increased when the enhancer is located upstream of the promoter. Okay. This is downstream. This is upstream. What are their rates? 4, 4. That statement is invalid. Out. A promoter bound with RNA polymerase and general transcription factors results in basal rate of transcription. Okay. Right. Promoter bound. Okay. D will dispute that because why? It's over here, right? And over here. Yeah, right. Promoter bound, yeah. Okay, so D will dispute that because generally you have enhancer here, you don't have anything bound to your promoter. Okay, it's having transcription over here. So that's why D is wrong. So B is the only thing that is okay. I'm going to tell you that is correct. The relative distance between the promoter and enhancer has no effect on the rate of transcription. When I look at E, E is in part B is near. E in part B is far. It's both 4, 4. So therefore, it's a valid conclusion. Okay. Okay, MCQs. Now for this questions on cell cycle, take note once you touch the deployed number 52, tell yourself, okay, when I go to anaphase 1, what happens at anaphase 1? My one chromosome was split into 2 chromatids. So if here is 52, after it's split, 52 times 2 gives me 104. So with this picture in mind, straight away life becomes very easy. Number of centromeres during metaphase, 1 chromosome, 1 centromere. So it should be 52. Number of chromatids, 104, correct? Number of DNA molecules in a cell at the end of anaphase 2. Now, at anaphase 2, has cytokinesis happened yet? No. So the amount of nucleotides, the amount of chromatids in that same cell hasn't divided to 26 yet. So answer is 52. Okay. So 26 is only after cytokinesis. And for those of you who have cell cycle in your promo, is cytokinesis part of mitosis and meiosis? It's not, okay, it's separate. Okay, good job. Okay, question 12 over here. Okay, following events occur in the extension of telomeres. So this one, I'm just simply looking at the order. First thing first that must happen is I have to have a complementary base pair with the RNA template. So here should be number 1. Now the trick here is this, they always give you a few options. Choose the option number 1 of all the options first, then you give it a try. If I start at 3, reverse transcription to extend, it can't start at 3. 4, complementary base pairs of the RNA template with single-stranded end of the telomere. This one can. So straight away, A and B is out. After there's complementary base pairing, what's going to happen now? Reverse transcriptase, the reverse transcription will happen. Then after that, the order just follows on. Ending part, DNA polymerase catalyzes the formation of a complementary strand. This is correct. Okay, so just use the hint here. What is number 3? What is number 4? Take a look at it. See which one is correct. Slowly eliminate. Okay, question 13. This one is on respiration. All right, first thing first, let's eliminate some options. V, this thing here with the round thing and the channel here has to be ATP synthase. So straight away, C and D are out. Over here, which one is the reduced coenzyme? So what are your reduced coenzymes? You have FADH2 and NADH. So FADH2 and NADH has to be R and P. So the answer here is P. Okay, it cannot be S. So here is gone. Answer is A. Okay, can I check with you? NADH will be R or P? Okay, the answer is NADH. We'll cover that in the respiration revision later. Okay. Now, question 14 over here. This is the example of choosing the least wrong answer. What do I mean? Okay, first thing first, I have pyruvate. Okay, I have anaerobic respiration in these cells. Pyruvate is being broken down. When pyruvate is broken down to ethanol, this is 3 carbon, this is 2 carbon. Where did 1 carbon disappear to? This is carbon dioxide. So carbon dioxide is indeed a waste product. So option 1 is correct. Now, let's look at option 2. Compound Y is necessary for more rounds of glycolysis. What is compound Y? So if we recall, what's happening here is NADH will be regenerated to form NAD+. Which one do you need for glycolysis? NADH or NAD+? You need NAD+. So this is wrong. So question 2 is wrong. Option 2 is wrong. That color. Then now, option 3. Compound Z starts functions as an oxidizing agent. If you really know 2 is wrong, okay, A, B, D, they're all gone. Option 3 technically is not really correct. Why? In chemistry, so ethanol to ethanol is a reduction reaction. If this is being reduced, which is the oxidizing agent? Y or Z? In chemistry. If ethanol is being reduced, which is the oxidizing agent? The compound before or the product after? The compound before, right? But here say Z acts as an oxidizing agent. So when I see, I was like, not very correct. But since I know 2 is confirmed wrong, And there's no answer here that says 1 only, right? Okay, you choose what is the least wrong. Answer here is C. Okay. All right, last one. Okay, different types of reactions occurring in your Calvin cycle. This is respiration. Straightforward. I'll just use this picture to guide you. First thing first, decarboxylation, okay, occurs when you convert RUBP to GP. RUBP to GP over here. Is there decarboxylation? No, you are adding a carbon dioxide. Straight away, this is wrong. Okay. Then B, the GP people look very stunned. Well, it's okay, no worries. Today fresh for you. Lucky thing I'm not going through photosynthesis today. Okay. Now, I just put this question here in case some of you got photosynthesis in your final exams or promos. Next, phosphorylation occurs in the conversion of RUBP to GP. Okay. RUBP to GP, there's no phosphorylation once again. It's just adding a carbon. All right. These are for the folks online who have photosynthesis and the people here. Check. Who has photosynthesis in a promo? Wow, nobody. Okay, let's go very quickly. Decarboxylation occurs in the conversion of triphosphate. Nah, this one doesn't happen. So it's carbon reduction. Okay, not carboxylation. So the answer here is just C. Okay, let's go to structured questions now. All still okay? All still alive? Now, structured questions, you don't have to copy too much of the answers. They are available online, but you need to look at the question analysis. All right, so first thing first, describe the process. Describe means you have to look at a picture. So if you all don't know, describe as a command word, okay, you need to quote figures or use evidence in figure. So what is the only evidence you can use here? It will be the English, phrasings here, cleavage lah, processing lah, anchoring. So this all is the English that you can use to prove to the marker you are describing. Now, first part. They ask you what are the events occurring between, okay, cleavage of the rough ER and export. So cleavage of rough ER here, export is all the way here. So straightforward, wow, I can talk about anything here. You can talk about, I will cleave the signal sequence. I will export the membrane for anchorage. There's glycosylation or proteolytic processing, okay. So there are so many things you can talk about over here. You just need to weave it into your answer. Then you must put in proper English. So what are the sequences of events? First thing first, after the RER, okay, vesicles containing your lactase will undergo glycosylation and cleavage of the amino acid signal. Where did I get that from? Here. Next, since it's going to move, okay, from the Golgi apparatus, it'll be packaged into Golgi, okay, vesicles, or it buds off from the Golgi. Next, this part over here, export and anchoring, I have to describe that part. So the vesicles will move and fuse with the cell surface membrane before finally having this particular portion over here. How do I get membrane anchoring? By saying it embedded, okay, to the embedding of protein in a vascular membrane. So your phrasing can be in any form, but these are the 4 ideas that should be there. Okay, so out of 4, 3 will do. Because this question is pretty generous. Okay. Now, answers are available online. All right, so I will not spend time letting you copy. Now, suggest, for those of you who don't know, okay, the suggest command word. Okay, let me ask. How did the school teach you what suggest command word is? What are the requirements of a suggest command word? Okay, so suggest means it's not in your syllabus. Meaning to say it's not in any of the learning objectives. Huh? How they test us? It means it's not in your learning objective, but you have been taught something similar. You are supposed to use that knowledge to apply it to here. So the greatest and easiest way to understand it is swimming. Where did all of you learn how to swim? Some swimming pool at your house, okay, or the swimming complex, right? Then one day you got class bonding. You go Sentosa. Hey, the water very nice. I want to swim in the beach. How come you can swim in the beach? No one has ever taught you to swim in the beach, but you learn to swim in the pool. That is called a suggest question. Because in your head, swimming pool or water, I move my hand, I move my leg, I don't drown. I suppose if I do the same in the sea, I will die, I won't die lah. That is a suggest question. So use stuff that you know already. And if you panic, just tell yourself this. They won't ask you a suggest question that they haven't taught you. You are able to solve it. You just need to think, what have I learned? So here, they're asking you, why must the signal sequence be cleaved to form the prolactase? In short, this thing is useless. If I can cut it off, it's useless. So why would it be useless? Think of it. How can I phrase it in the answer? So this signal sequence, before it forms the proform, okay, it's useful. After I form the proform, not so useful anymore. So it's no longer required after the protein is directed there. Okay. Then another way could be, I need to process this. If I need to cut this lactase later, and I have this signal sequence, it might be blocking. Okay. So it might be blocking the enzyme that's supposed to come and cleave it, physically blocking the thing. So that's one other possible answer. With those answers, can I check anyone has any answers that you want to try? That's point number one. Good job. Thanks for your answer, Kanishka. Anyone else? Okay. Okay. Not, I'll move on. Okay. Now this one over here, wow, very complicated question. In short, I have to look at color, gray color, black color, and I have to look at all of this. First thing to do to make it very easy, put down here by the side. They have told you it's modified cytosine. What is modified cytosine? It's simply DNA methylation. Once you know about DNA methylation, life becomes easier. When DNA is methylated, is that gene supposed to be expressed more or expressed less? That's right. So lesser gene expression. And once you understand this, this question is so easy. You have sliced through all the confusion like a hot knife through butter. So obviously now you know lah. If I have very, very silenced DNA here, I will have very low transcription. This just means transcription. So this is high transcription, this is low transcription. Then you just check the color lah. If I have very high methylation over here, I have very low transcription. Life is good. Then the question very straightforward. Okay. All right. Now over here, they're asking you, infant, adult milk. Okay. I have a son. He's 2.5 months old. He's at this stage. Fella drinks milk like no one's business. So he must be able to digest lactose. So it means all of the lactose genes, okay, need to be highly expressed, which means I should see a lot of white, which I do. White, at the back part a bit more black. Compared to an adult like us, there's a lot of gene expression, gene methylation here. Yes, you're right. So very clear difference. Babies drink milk because that's all they can do. They need to be able to digest milk. Given a choice for dinner tonight, will you drink milk or will you go and eat a McSpicy meal, which is at $6.50? One idea, right? In case you don't know, McSpicy meal is $6.50 for this period. McSpicy, right? McSpicy for lactose? No. So because as an adult, we don't have much lactose in our diet, that's why the gene is being silenced. Not like, now I know why I'm lactose intolerant. Yes, correct, because of this. Okay, let's go to the question. Now, they ask you, describe how the expression of LCT is influenced by environmental factors and age. They give you such a big hint. Environmental factors, age. So, like what I mentioned, as you get older, more genes get silenced. And there is a proximal and small, distal and small. So, distance plays a part as well. So, what are the answer? Comparatively to a young kid, the adult has a lower level of lactase expression. That's what you can see over here. A lot of gray, some white. Here, it's all white. Okay. Next, adults show a moderate level of expression in the proximal intestine. So you can compare, okay, locations. Infant at the proximal, there's a lot of expression. Adults at the proximal, a lot of silencing, very, very low levels of expression. Okay? Okay? Right. Then third one, expression of lactase in adults is higher when milk is consumed as part of the diet. How do I get this? For environmental factors, I'm looking between these two pictures. So now is the time to spot the difference. What is the difference that I see over here? Mainly, I can tell that after an adult has milk in the diet, this part starts to change. Here is lighter gray, here is darker gray. So I just write that as an answer. Okay, so I hope I just showed you that any kind of difficult question, a figure will give you the answers. So don't panic. It's just looking out for clues. Okay. It's mentioned the answers will be online, so I won't spend too much time letting you copy. Part 2, explain how the downregulation of gene expression occurred. So because they have mentioned it is about cytosine modification, it means it's DNA methylation, you start with that. So it's the whole chunk lah. DNA methylation. What happens when DNA is methylated? Okay, then together with DNA methylation, there's also histone deacetylation. So both of these were coming together. Okay, so first part talk about percentage of methylation of cytosine. Okay, is higher. Then what happens when this happens? It will attract a lot of histone deacetylase. Histone deacetylase will do its job. It will remove the acetyl groups. And when it removes the acetyl groups, your DNA becomes more negative, no, becomes more positive charge. With more positive charge, it will bind more negatively to the histones. Okay, your DNA will bind more to the histones. They'll become more tightly wrapped around each other. Your RNA polymerase cannot access it for transcription. So those of you who are marking your work, okay, I do believe the biggest part you will have missed might be part number 1. Okay, I know here and there you will write methylation and deacetylation, no issues. But part number 1 probably is not there. That's my guess. Oh, okay, Gwen's still alive. Yeah. Now, next question over here. This one becomes, wow, from the question number 1, now question number 2 a bit more easy, breathing space. For your promo exams, your teachers won't give you a lot of like 10 hard questions. There'll be some easy ones, there'll be some challenging ones. So if you want to strategize, which I don't recommend, but flip through the paper, look for the easy questions first, make sure you score it. What is easy is relative to everybody. It really depends on what you studied better the weeks before. So don't just start paper, look at question 1, do all the way. Scan through the whole paper. Look at which questions you know you can get the marks, do it first. Okay, you don't need to finish everything to get an A if you're aiming for A. You just need to hit 70 marks. Wow, you see until very easy. Okay, just telling you, scan through your paper, do the questions you can do first. Right. Now, over here, organelles in the mammalian cell. For such cell ultrastructure questions, my big tip to you is check what is the nature of the cell. If it's a plant cell, take note. If it's an animal cell, take note. Why? Some students don't take note of that. There's a question here that says animal cell. Some questions ask you about something, some organelle. Answer, chloroplast. Die liao lo. How to have chloroplast inside animal cell? Okay, don't laugh. I've seen quite a few batches of students doing that. And for my experience, that 1 mark lost from their mistake is usually the 1 mark needed for the kid to jump from 49 to 50, 59 to 60, 69 to 70. So it's always a very painful 1 mark that is lost that way. So be careful. So here, quite clear cut. Animal cell, double membrane. Now, once you see things moving into the middle here, right, that's the big hint. This must be an internal membrane. What is the only organelle that animals have that has an internal membrane? Mitochondria. Thank you very much. Okay, Irana. All right, then why A looks different? A is simply also the mitochondria, just that my water bottle like that, versus my water bottle like that. Just different orientation. This is B. This is A. That's all. That's how you cut it. Okay? So that's why it looks different. So those of you who say no, it's not the same organelle, I'm quite curious, which organelle do you think it was for A? So both of them are the same organelle, the mitochondria, mitochondrion. They ask you to explain, so give elaboration. Like I said, I can see the inner line there. I use the inner line to help me. The inner line is the cristae. Okay, So I can see the inner membrane. Then one question kids like to ask, how can you tell that is a double membrane? If you look very closely, especially at this segment here, can you see there are two lines? So you will be able to see a double membrane in a cell picture if you look at the correct spot. So here, two lines, double membrane. Okay. Then over here, if I look at this is the RER. How do I know? Yes, spots there. Spots are ribosomes. So one tip for you, for such pictures, every time you see a dot in a cell diagram, it's a ribosome. Next, back to the whole thing about membrane. Where the dots are, you can see there are two lines. So, two lines here, it's a single membrane here. Here got one line, one membrane. Inside, got more lines. So, two membranes. So that's how you can tell the picture what is a double membrane, what is a single membrane. Okay? Okay. Truth be told, I have no idea what it is. So luckily, they never test me. Now, moving on. During maturation, red blood cell loses its nucleus and most other organelles. In short, it becomes a container. It has no nucleus, no ribosome, no RER, no lysosome, no mitochondria. Is it even alive? I don't know. Good question. Okay. So what they do all of this to have space for hemoglobin molecules. Fun fact, each red blood cell has about 300 million hemoglobin proteins. Okay, you will not be tested this. I just found it out. So over here, they tell you, no mitochondria, still can synthesize ATP. Once you know that there's no mitochondria, if I look at respiration, which are the steps that involves the mitochondria? It will be your Krebs cycle. Okay, and your link reaction. So this is in the mitochondria. If I have no mitochondria, straight away those are out. It should not even be my answer. But if I can synthesize ATP, the whole topic of respiration is to synthesize ATP. I have my glycolysis, link reaction, and Krebs cycle. All three, okay, directly or indirectly will synthesize some ATP. So I'm left with glycolysis. So it has to be glycolysis. Why? Okay. I can use glycolysis, okay, to produce 2 ATP per round of glycolysis. But can I do it alone? No, I have to regenerate my NADH. And that's where fermentation comes in. Okay, so if you just write anaerobic respiration, also correct. Also correct. Okay, so for 3 marks, first mark will be you mentioned that anaerobic respiration. Glycolysis can happen with anaerobic respiration, so it will produce 2 net ATP per glucose molecule. Okay, why? Because with my lactate fermentation, I will regenerate my NAD for glycolysis to occur. So once you know that there's no mitochondria and you have eliminated these two, you can tell yourself, I just need to focus on glycolysis and anaerobic respiration. Okay. So far the questions, look very difficult, but once you break it down and you analyze it, it's actually more manageable. That's what I hope one thing you can take away from today after the mock. Okay, question C over here. Same thing as MCQ, you can color code it. I have 3 different chemicals. I have 3 different effects on the mitochondrion. I have this very chim thing over here. It tells me it measures my oxygen consumption rate. They tell me I've added these different chemicals at different timings, and it therefore gives me this graph. Now, first question they ask you, why does oligomycin cause a drop in my OCR? Let's look at what oligomycin does. Now, oligomycin inhibits ATP synthase. Okay, so if I inhibit ATP synthase, if I were to draw my ATP synthase like this, okay, now it's stuck. What is supposed to flow through my ATP synthase if it's functioning properly? H plus, thank you so much. Okay, what was that? Thank you so much. H plus, so protons supposed to flow through. So if my protons cannot flow through, what happens now? It's kind of like you when you have constipation. If you cannot go to the toilet, then you're stuck here. You feel that, a lot of pressure, What happens? You get very uncomfortable. Okay. Things are only restored once you go to the toilet. So same thing here. If my H plus cannot flow through, the high proton gradient in the intermembrane space will continue to get higher and higher and higher. when it becomes so high, can any more protons flow up? All right. If the protons, cannot flow up, okay, it also means that the electrons that are passing through the ETC, can it flow through the ETC? Cannot. If it cannot flow through the ETC, what is the final electron acceptor? Oxygen, right? If electrons cannot flow through, do I need more oxygen? No. Let's look at the oxygen consumption rate. It decreases. Make sense now? Because there is no need for oxygen. Because electrons cannot flow through the ETC now. Because the whole proton gradient is so high, things are stuck there. And that's your answer. Okay? Electrons cannot be passed down the ETC. Okay? To reach the final electron acceptor, oxygen. For 2 marks, 2 key ideas. Okay? All right. Okay, I hope you noted down my analysis. Next, part 2, FCCP. What in the world does FCCP do? Okay, FCCP causes protons to leak out of the mitochondrial intermembrane space. Okay, leak out of the intermembrane space back into mitochondria. In short, it means if I draw the thing like that, H plus is just flowing back through here. So once again, H plus flowing back through the membrane when it's not supposed to. What happens to the proton gradient? It will be affected. It will be less steep as compared to before. So if it's less steep as compared to before, my electrons can flow even more. Because why? Proton gradient is very little. More H plus can go up. So more electrons can continue to flow and flow and flow. If more electrons can flow, I need more oxygen as the final acceptor. And therefore, the oxygen consumption goes up. Okay? And that is the answer. So the whole focus on these 2 parts is really the fact that can my electron flow? High proton gradient, low proton gradient will affect the ability of H plus to be pumped up. If H plus cannot be pumped up, electron flow will be affected. Okay, then link it back to oxygen as the final electron acceptor. Now, part 2 over here is the teacher trying to test you math. It might come out. For this, let me give you one very big hint. Do working. Why? If the question here is 2 marks, 1 mark is for working. That's how low the bar bio teacher set for math. Okay, so do a working also to confirm yourself. Right, and how do I get this? They asked me to find out the leakage by determining the difference in the OCR at 20 minutes and after adding 20 minutes. So just look at the graph. It will be at 57 minus 30. So 20 minutes after adding oligomycin, 20 minutes after adding that. You find the value, minus together, very straightforward. Okay, question 3, replication over here. Let me remove, see. Now, what is the enzyme responsible for opening the replication fork? This one straightforward lah, helicase, you all know this. On 3.1, label the leading and lagging strands. So just take note, lagging strand is always away from bubble, away from fork. Okay, it's away from the fork. Leading strand is towards the fork. Okay. Right. When you draw this, please use pencil. Don't you draw in pen. Next, they ask you why DNA replication requires both leading and lagging strand synthesis. So later I'll cover this in greater detail. But basically, if I'm opening it up over here, every time I open more and more, okay, my leading strand can just continue all the way. Life is very good. But once I've opened up this part of the lagging strand, okay, or let me draw more. Once I open up this part in the lagging strand, because replication goes in the opposite direction, who is supposed to replicate this? You must wait for the next primer to add something here, then it can start replicating all the way. So that's why it's very slow. Okay, that's why it's very slow. So, yeah, the first part. Okay. Then next thing is my leading and lagging strand both were synthesized a template strand. and I cannot afford, my leading strand just synthesizes one strand, is done. I'll be left with 3 DNA strands, which means I will have an incomplete DNA molecule. So the whole idea why both must happen at the same time is so that at the end of DNA replication, I have 2 DNA molecules, both with 2 strands. So that is the direction this is going to go towards. Okay. Then it's also because it's anti-parallel. So if all those coming together, okay, it can only replicate in an anti-parallel manner in your 5' to 3'. Okay. Then why would there be the leading and lagging strand? Lagging will give you your Okazaki fragments. Now, pro tip over here, how to remember this. Okay. Now, those of you who have already learned protein synthesis, when you synthesize your mRNA, then after that they got the post-transcriptional modification, right? What's the first modification that happens to the mRNA? What's added to it? 5' cap. Thank you very much, Satya. So if I can add my 5' cap, it means the 5' cap must come out first, right? Whether is it transcription, which produces RNA, or DNA replication that synthesizes DNA, it is the same direction of synthesis. So you can remember that using your 5' cap. So 5 to 3. If you find that very complicated, I can remember the opposite manner. This is direction of synthesis. What is the direction of reading? It will be 3 to 5. So you say very easy. Exam, I cannot remember. The exam paper will give you the answer. All you need to do is look at your exam paper. Now, when you read your page number, do you read from low? You read your book, you read from the earlier page, smaller page to larger number, right? Does anyone read from the last part of the page backwards? If you are special. I'm not. But basically, you always read your pages from the smallest number to largest number. Same thing. DNA will do the same thing. It will read it from a 3' direction to a 5' direction. Okay? So I hope that helps you a lot. Now, after that, draw the newly synthesized strands. The only issue here is just make sure that at your 3' end, there is a gap. Because why? This will form your end replication problem. So if you're happy, wow, you get to draw all the way until here, boom, shakalaka, is wrong. Okay, so take note, your 3' end has to have a gap. What used to be there? Jolene. Sorry. What used to be here? This used to be your primer. Okay, this used to be your primer. Okay. All right. Oh, right on schedule. Okay. And over here, compare DNA polymerase and telomerase. This is a very commonly tested question. Yeah, Jia, you want your copy? Okay, very good question. Thank you so much. So, Xing Ying is asking, how come for the leading strand, there is no end replication problem? So, firstly, let's look at the direction of synthesis. Okay, this is 3'. I'm reading this way. So, this strand, Xing Ying, is being synthesized in this manner. Agreed? So, if I'm synthesizing it in this manner, where would my primer be? My primer will be somewhere here. So, when it synthesizes all the way, if a primer like this, my RNA polymerase in purple will bind here. It can go all the way to the end. So once it reaches the end over here, would I remove anything here? Is there a primer there? No, there's no primer. But on the other end, over here, when we look at the opposite end, the lagging strand, I put a primer here. It synthesizes in this direction. My DNA polymerase will go here. Agreed? After it has done its job, reach all the way here, life is good. The primer will be removed, right? I remove it. Once I remove the primer, there's a gap. So that's why this one has a gap. The leading strand doesn't have a gap. Yeah, okay, so the primer excised here, the space that it was at would have been covered up by the other synthesis of the leading strand behind it. So for here, the issue is there's no strand here to synthesize to cover up this gap. Okay, good question. I'm sure quite a few of you also probably have this question. So I thank you for your courage asking. All right. Anyway, later I'll cover that a bit. Okay, part E. Whoa. Okay, now when you revise your different polymerases, RNA polymerase, DNA polymerase, and telomerase, be very clear they can ask you some compare question. So to help yourself, straight away I'll ask myself, what does DNA polymerase read? It reads a DNA template. What does telomerase read? Also DNA template. Okay, sure, so I can't use that. But what template does telomerase use? Is it also made of DNA? Your telomerase uses a RNA template. So once you know, hey, got RNA template already, that can be one of your main difference. All right, next, compare question. Do I need only differences, only similarities, or do I need both? Both. So one of the very common mistakes is, okay, this one needs both. So it's either 3S, 1D, 1S, 3D, or 2S, 2D. D for difference, S for similarities. And in this question over here, I would generally go for more similarities. Because the only difference I think at your level that you can easily remember would be this, the template. So what are the similarities? Go for very simple stuff. Both synthesize DNA. Both involve nucleotides. Okay? So got complementary base pairing. Both were synthesized in a 5' to 3' direction of the new strand. You want to say both involve 3 deoxyribonucleotides, also can. Both extend DNA, also can. So there are a lot of answers, but don't repeat the same idea. Now, what are the differences? Differences over here, all right. I will go with template. DNA replication uses the DNA template. Okay. Telomerase uses the RNA template. Right. Then base pairing for DNA replication is DNA to DNA. Telomerase is deoxyribonucleotides to RNA. Okay. Then, okay, for presence, DNA replication happens in all replicating cells. Telomerase, for those of you who don't know, okay, only happens in your stem cells. So cells without telomerase are supposed to die, your normal cells. Okay, okay, so if you ever learn stem cells, that is fine. Okay, question so far? Okay, we're ending soon, soon we have a break. Okay, this one over here on the neck. Okay, why I put this question here, very straightforward. You might be asked to do this one day, either in MCQ or in structured. Please take note of the direction. So you must draw your answer and put the direction. This is an examiner remark. So what's the examiner remark? If your teacher hasn't told you, teachers have access to the complaints that the A-level markers have. They will write a report on how the Singapore cohort did and say what are some common mistakes. They have mentioned before that for such questions, students leave out the numbers. So you must write the numbers because the numbers confer direction. So I don't understand what you just said. In a simple nutshell, if you don't have 3, you don't have 5, but you have TAG, GCA, you get 0. Okay, okay. Wow. Okay, Natalie, I'll clarify it in the break later. Okay, okay. Finishing soon, finishing soon. I'll come back to your question later. Now, list one way in which the structure of an mRNA molecule is different from the structure of a tRNA molecule. For this, let's look at some pictures. This thing has so many juicy stuff to talk about. You're watching first. Which one is tRNA? Left or right? This one right, some tRNA, okay? Right, tRNA. So what does tRNA have? Codon law. Okay, so not codon. Anticodon law. Okay, it has an amino acid attachment site. Okay, structure-wise, what can you tell? It is not flat. It's folded onto itself. It can form internal, okay, complementary base pairs. This one is flat. This one, what do I call 3 nucleotides together? I call it a codon. Okay, so things like that will come to mind. Then they ask you to list one way which is different. The easiest, lowest hanging fruit is this. One got codon, one got anticodon. Easiest, lowest hanging fruit. Okay. Right. You want to go for more chim stuff, you can say one, okay, forms this clover leaf structure, one is linear. Okay, but to me, most of you should write the first one. Easy. Question so far. Okay. Now, so over here, this suggests how two structural differences identified in B contributes to the role of the tRNA protein synthesis. Now, your tRNA protein synthesis, okay, your tRNA only has one main role, okay? It is like your grapefruit driver. What does your grapefruit driver do? Make sure, thank you, Satya, say. To where? Sounds like, okay, let me explain on that. So Satya said, deliver food to us. Specifically, let's say if Satya, what's your favorite food? Whatever I get, okay, time to give you some army biscuits. Okay, let's say Satya ordered the McSpicy meal from McDonald's later, okay, through Grab. Then, he reached out, ready, McSpicy, $6.50, I saved $1.70, that's the offer. You open up, how come it's a filet of fish? Straight away very sad. Why? Grab driver, deliver McDonald's. What they deliver? The wrong thing. So, one aspect that students forget about tRNA is it must deliver the correct amino acid. And how does it ensure that it is the correct amino acid? The amino acid that is attached here must match the anticodon. Same thing, your Grab driver must deliver the McSpicy meal to Satya from McDonald's and not the Glendon Phua meal to Satya. That was the filet of fish one. Okay, can? And with that, okay. So key things, complementary base pairs, okay. So presence of anticodons for complementary base pair to the codons so that it codes for the correct amino acid. Exactly what I said. Make sure I get my McSpicy burger, not filet of fish. Okay. Next thing is this. If I need to attach an amino acid, I must have one part to hold on to the amino acid. Same thing as a grapefruit driver. How do they hold your food? They don't hold it in one hand, right? They got a very nice bag, square, very thermal. By the way, that bag costs $200. It's very good. Okay. How I know? My brother did it before. So we look at the back. A lot of project work has gone to this. A lot of prototyping, a lot of asking experts. So it's really very good. So same thing, your amino acid has to have this 3' end. Sorry, your tRNA has to have this 3' end to accept the covalent attachment of your amino acid. So two parts, bringing correctly, bringing the right thing. Okay. Then part 3, this one quite straightforward. Describe one type of post-transcriptional modification done. This one, once you see pre to mature, most students will write introns to exons. No issue, keep it up. Okay. Right. What I like to write, the easier one, okay, is adding of the 5', okay, cap. So my mature mRNA got 5' cap, pre-mRNA don't have 5' cap yet. If you're very confused, then what is considered as pre-mRNA? It is really that naked mRNA that just left the ribosome. Nothing has been done to it yet. So it's just purely ribonucleotides. Once you add the 5' cap, 3' poly A tail, and you do the splicing, it's in the process of becoming a mature mRNA. Yeah. And with that, I believe I'm done. Okay. Good job, everyone. Let's go for a short 5-minute break. Natalie, I'll answer your question now. Yes, Natalie, you're right. Okay, DNA polymerase and telomerase both catalyze phosphodiester bonds between deoxyribonucleotides. Yes, that's correct. Difference, DNA polymerase synthesizes non-repeating DNA. Oh, that's an interesting point. Okay, yeah. So the part about for telomerase is synthesizing repeated sequences. I'll give you a mark for that. That's a very novel point. Good job. DNA polymerase requires RNA primers to synthesize, but telomerase does not. That's correct as well. Okay, thanks for your answer, Natalie. All three are correct. The level of detail is impeccable. Keep it up. Okay, so for those online, let's go for a short. Okay, your break will be the length of this song. You're like, what the? Don't worry, I will use this to teach you bio later.
너를 위해 구웠지. But you know that it ain't for free. Yeah. 내가 만든 쿠키 너무 부드러우니 자꾸만 더 오르니 널 초코칩으로 스프링클러 입맛 버리게 만들고 싶어 숨기고 있지만 날 다 보고 싶어 If you want it, you can get it If you want it, 네 목소리를 더 들려줘 boy 식사는 없어 배고파도 음료는 없어 목말라도 달콤한 맛만 디저트 맛만 원하게 될 거 알잖아 식사는 없어 배고파도 음료는 없어 목말라도 달콤한 맛만 디저트 맛만 원하게 될 거 알잖아 내가 만든 쿠키 너에게는 독이지 네 마음속을 녹이지 Yeah Looking at my cookie 역시 향기부터 다르니 한 입은 모자르니 널 초코칩으로 스프링클러 정신 못 차리게 만들고 싶어 숨기고 있지만 나도 보고 싶어 If you want it, you can get it If you want it, 네 목소리를 더 들려줘 boy 식사는 없어 배고파도 음료는 없어 목말라도 달콤한 맛만 디저트 맛만 원하게 될 거 알잖아 식사는 없어 배고파도 음료는 없어 목말라도 달콤한 맛만 디저트 맛만 원하게 될 거 알잖아 Bet you know, bet you know, bet you know 레시피는 없어 딴 데서는 못 찾아 Take it, don't break it I wanna see you taste it Sugar, got sugar 난 재료 안 아끼지 Bet you know, bet you know, bet you know 네 다이어트 망치고 싶어 나 Take it, don't break it I wanna see you taste it Sugar, got sugar Bet you want some 식사는 없어 배고파도 음료는 없어 목말라도 달콤한
What are they doing now? Ah, remember that. Later remember that.
맛만 디저트 맛만 원하게 될 거 알잖아 식사는 없어 배고파도 음료는 없어 목말라도 달콤한 맛만 디저트 맛만 원하게 될 거 알잖아 내가 만든 쿠키 Come and take a lookie 우리 집에만 있지 놀러와 얼마든지 굽지 그런데 나 충치 생겨도 난 몰라 내가 만든 쿠키 Come and take a lookie 우리 집에만 있지 놀러와 얼마든지 굽지 그런데 너 충치 생겨도 난 몰라
Okay. Hi, Matilda. Can I say that both DNA polymer systems are enzymes for similarities? I would say yes, you can get the mark for that, but it's a bit I would say keep that in your pocket if you really cannot think of any other answers. So if you have three other better difference or any other better difference, no, three other similarities which are better, I'll put it first. I'll keep this as a reserve lah. Because my worry is your teacher might say it's too simple, but you are factually correct. So at A levels, they'll give you the mark. At promos, your teacher's job is to try and train you to be better. So they might want a bit more higher level answer. But you are factually correct. Can Matilda? One more question. Okay, welcome. So polite, all of you. Okay. Now, can I invite you to take out your notes? Okay, everyone at home, I hope you are back. We are missing Nidhi, Kanishka, Hiranya. Oh my God, I'm massacring her name. Oh, so I can check. Have you all managed to see any of our advertisements on IG? What? You got see? What? Wow, IG is doing their job. Good job. Okay. So when you all see the ad, what do you all do? Do you all skip right, like, or you all click enter? Find out more. No, honest question. Because when I see all the other tuition companies, I just next, very fast. IG is saturated with so much tuition content. Okay, now I'm going to start this part. Do take note, later you'll be, why all his slides don't look like what is given to us? What I'm teaching you is part by part in slides. What you have on your paper is the same content, just that delivered in a different way. Okay, so my main aim for the 3 topical revisions today is such that at the end of the day, you are able to understand things better. You are able to remember the difficult parts in a less painful way. And at the very least, when you see this topic again for promos, you are more confident in doing it. Okay. All right, if not, let's start firstly DNA replication. So can I invite you to turn your pages, I think, to page 18 on DNA replication. Oh, another question. You know the cakes there, the London Choco Roll, all this. Do you like that? Like that? Wow, why? Sorry, not judging, but why? And I saw that, studying my option, like, hey, can you? I think the kids don't like this, but every kid I asked, like Gabriella, she's like, yeah, it's the best thing ever. Yeah. Good. Please eat more. We have a lot of that. Okay, so firstly, let's start with the structure of DNA. I'll cover some pointers over here. Oh, that's really fast. Good job. Do you all know each other or you all are now best friends? Now everyone's like, oh yeah, good idea. I should have bought milk and hey, where's the McSpicy? There's one really far. Like there. Nearby. Okay. Let's start. Now, this is the structure of a nucleotide. Very straightforward. I need you to know this whole thing is called a nucleotide. If they ask you to label, okay, the pentose sugar, but in your structured question, they have given you somewhere the name of one nucleotide, like adenine, thymine, cytosine, whatnot, right? ATCG, right? Please don't write pentose sugar. So I say again, if they ask you to identify this particular thing over here, okay, and they have given you adenine, thymine, cytosine, guanine somewhere in the question, right? You cannot write the word pentose sugar. You must be as specific as possible. So if here is A, okay, and they ask you what is this base over here, it should be thymine. Okay, sorry, for nitrogenous base, not your pentose sugar. Sorry about that. For a pentose sugar, you shouldn't say pentose sugar. If you know this is a DNA molecule, what is this specific pentose sugar called? Deoxyribose. Thank you very much, singing. Okay, so I say again, don't call it a pentose sugar. If they ask you to label this pentose sugar, your answer should be deoxyribose. Okay, this is the examiner report. Now, why is pentose sugar not accepted? In chemistry, what is a pentose? Anything that is 5 carbon or 5 ring. Okay, so there can be a lot of things that are pentose sugar, but deoxyribose is a very specific molecule. So write deoxyribose. Okay. Okay, next one. Pain to most of you. Sure, I know got the adenine, cytosine, guanine, uracil, thymine. How to remember which one is the 2 ring, which one is the 1 ring? Now, there's a very simple one at the bottom. CUT is the 1 ring. Okay, that one I don't know whether you can remember. Let me give you a different way on how to remember which particular nucleotides are double ring. So here's the story. Long ago, I was talking to my senior. Okay, caveat, this is a really stupid story. But I'm sure at the end of the day, you'll be able to remember something that has two rings. So like this senior, she was telling me, okay, he liked this girl. Okay, so story goes like this. Adenine sounds like the name Adeline. Guanyin sounds like what female name? Okay, Geraldine. A bit far-fetched, but no, it's okay, it's okay. And that's it's a stupid story. So here the story goes. So at this senior, okay, he liked this girl called Adeline. So ask him, hey, how you remember the purines and pyrimidines? You're saying, oh bro, a very easy one. I can this girl called Adeline. So because Adeline has very nice big round eyes, and she has two eyes, right? Don't spit. Hey, spectacle, what? Anyway, the fictitious person. I just Google AI girl, come out image. Okay? So after he said that, I cannot forget the idea that the girl he liked called Adeline. Adeline has very big spectacles, two ring over here. So anything that Adeline got two ring, in short, any of the nitrogenous bases that sounds like girls' names, I'm reminded of the fact that a girl has big spectacles like this is purine. Okay, it makes zero sense. But I'm sure tomorrow when you wake up, damn it, it's still in my head. Okay, so that's how I remember lah. Adeline sounds like adenine, Geraldine sounds like guanine. The names of the nitrogenous bases that sound like girls, think of a girl with a big spectacle. Okay, we have quite a few here in the classroom actually. Okay, right? Now, if you ask, doesn't that work for boys? I've never explored that idea. So moving on. Next thing, okay, so what do you need for DNA replication? Some things are location is always in the nucleus. Why? That is where DNA is. Okay, and for everything else, to enter the nucleus, you go through the pores. You will see this word here, activated nucleotides. What is that? Don't worry. In your answer, you can just say nucleotides, it's fine. If you see activated, it just means energy has been supplied to it. Energy from what? ATP. Okay, right? Okay, so I will tell the story of DNA replication kind of like how you try to fix a jigsaw puzzle. So when I buy a jigsaw puzzle, what does your jigsaw puzzle come in? A bag, right? You open the bag, there'll be two ends. So same thing like your Ori. When I open the double helix, I separate it. Okay, it's like opening a Ziploc bag. I have two ends at the side over here. Someone's facing, so impressive, good job. Okay, so first part, to separate it, what is the enzyme involved? Helicase. Students generally, from my experience, have no issue remembering this. But now let's imagine, my DNA double helix is really very tightly wound into a double helix. When I separate it, okay, with helicase, will it stay there? No, right? So once I let go, you'll go back. Kind of like if I undo my hair now and I put my man bun hair here, if I don't hold it up, you'll keep covering my face. I was very tempted to do it, but I scare you off and you'll call police. So I should not do that. Okay, so after you separate it, it must be separated and it must stay there. So what holds it there? Single-stranded binding proteins. Okay, so in your head, it's kind of like after you open the back of jigsaw puzzle pieces, you must keep the back open. Now, you will form this thing called a Y-shaped replication fork. Why they use the word Y-shape? Because it looks Y-shaped. So your helicase will always open towards the zip. And here is where we call the Y-shape. Next part. So it will always go in both directions. Why? Because time is money and your DNA doesn't have the time to just go in one direction. Anyway, one direction also doesn't exist. So that's why two directions. So over here, that's like a ziploc bag. When you open your ziploc bag, it will open on both sides. Okay, so that is why you always have two replication blocks. And if this comes up in the MCQ, please take note. You only have one replication bubble at a single origin. One replication bubble has two replication forks. Okay, so this whole round thing here is called a replication bubble. The forks are by the side. Okay, this is quite a common A-level question, in all honesty. Now, once we have separated them and we have started replication, that is where the headache comes in. Lagging strand, leading strand. Okay, so for that, let me focus on the half of the replication bubble. Now, you know by now, before I can start my DNA replication, I must add something there. I must add my primer. Kids always ask, why? Very simple. You all got watch the recently concluded Olympics. You all got watch the 100-meter sprinting event. Well, what you watch. Okay, you know, you watch sprinter sprint, they will put their leg on some standing block. Why? To make them go faster. If you don't put the block there, technically they can run. But I see the primer kind of like the standing block. DNA polymerase needs to shoot out really fast to replicate things very quickly. Why? Your DNA is kind of long. So to help it bind there faster, it needs a starting block. What is the starting block? Your RNA primer. Okay, so think of the primer as a starting block. What will synthesize the primer? Primase. Okay, now, so at this stage, if I'm going to synthesize the primer, my primer is a ribonucleotide with phosphodiester bonds between them. Please learn the word adjacent. Okay, not 2 Jason, not 3 Jason, but adjacent. 1 Jason. Okay, you all didn't laugh at my code joke, so sad. Okay, never mind. I have a bio teacher here called Jason. I keep telling him, bro, you must crack that joke. Jason. Okay, so now, once again, how to remember how DNA template is always read? It's just like reading a book. You read from 3 to 5 pages. Next part, which might come out in an MCQ question, or if your teachers are very mean, short question, 1, 2 mark. How come? How come my DNA polymerase needs a primer to start? Now, to simplify this whole thing, DNA polymerase is what type of molecule? It's an enzyme, right? You all learn in bio. Enzyme, what's the most important part of an enzyme? Active site. Thank you very much. Yes, finally, Mr. Paul asking something I can answer. Okay, active site. So, think in very simple terms. Your DNA polymerase has a very specific active site. In order to form the shape where it can bind to on the DNA, so DNA is very thin lah. So DNA is like that, so thin. Your DNA polymerase is so big, a lot of empty space here, right? So in order to fill up the active site to make it big enough, hold on, let me bring a top. I forgot got a top for this. My orange. Okay. So DNA very thin. Polymerase in so much space there. I put the primer here, what happened? Wow, relatively better shape. So once you see it from this angle, it's just an enzyme and complementary shape. Yes. Then I contextualize it to the question. If I have the RNA primer there, what does an RNA have that gives me the complementary shape? It is this specific thing, a 3' OH group. Then you might ask, why got 3' OH group? Eventually, when I elongate this whole long strand, how do I join all the nucleotides that are adjacent? I'm forming phosphodiester bonds. Right? What do I need to form phosphodiester bond? There is OH. So by supplying a primer there, your DNA polymerase, I have the first OH to form my phosphodiester bond. You are right. That's how you have the complementary shape as an enzyme. Okay, so I realize I'm teaching it this way, kids become less scared of it. I'm sure your school might have taught you another way. That is the theoretical way. That is correct. But not many schools spend enough time explaining it in the enzyme perspective. Okay, okay. All right. In terms of jigsaw, okay, if I were to make a jigsaw, okay, I don't know how many of you have a habit of making jigsaw. You will always start with the corner piece, right? The corner piece will start you off for the whole entire jigsaw. Your primer can also be said to be like a jigsaw corner piece to start the whole entire jigsaw puzzle from being built. Okay, so this is in picture whatever I was saying. Once again, DNA polymerase has a complementary shape at the active site. It must recognize this specific OH over here. Then it can bind. After it binds, then it can start replication. Okay. Now, synthesis of daughter strand read in 3' to 5', regardless of wherever, is always 3 to 5'. So this is something you have to learn as a student. 3 to 5' is a relative thing. If I am given one corner, 3' here, can you see this? You must surely deduce, the opposite end here is 5. Then if I draw any strand, wherever I stop, as long as I started at 5 here, wherever I stop will be 3. Because this 3 is relative to this 5. A lot of kids get very confused. If here is 3, then why here is also 3? This strand here is 3 because this is a 5. And relative to this 5' end, here must be the 3' end. Okay. So one thing that can come out in the exam is they will give you a diagram with a DNA molecule, bend until like your iPhone cable. Okay. Then they just label one end 3'. you are able to come up with the direction just by tracing them. Okay, so just remember, once you know one is 3 prime, the other one must be 5, follow it all the way. Okay. So as you can see, the blue line above is the reading. And as I said, you flip your pages, you read your book from the lower page number to the bigger page number. But in the reverse direction, the new daughter strand will be synthesized from the 5 prime end, the 3 prime end, because of anti-parallel nature. Okay, so once again, like reading a book, you read from the smaller page number to the bigger page number. Now, so the complementary based pairing is kind of like a jigsaw puzzle. Once I have one template strand, everything else, just like jigsaw, I will put the matching piece. Okay, so it's quite straightforward. What I want to say here is this. One thing here, like this whole entire long strand is called a strand. The new daughter strand is called a daughter strand. Two strands together, what do I call it? A molecule. Okay, so individually, this is called a DNA strand. Two DNA strands form a DNA molecule. That's another common mistake students make. This English part. One strand, second strand together form molecule. Next question. So once I use the word parental template strand when I explain this, don't need. Just say template strand all the way. The teacher will understand. The daughter cannot be a template strand. Only the parent strand can be a template. So parental template strand is a bit useless. Okay. So I'm saying I'm a boy. No, it's like saying okay. Next, over here, okay, catalyzation of phosphodiester bond. This one, just take note, is 3 prime to 5 prime. Okay. Okay, so as my DNA polymerase reads the thing, it will also synthesize and add new nucleotides. Please take note, when you're explaining what DNA polymerase does, in biology, as long as you're talking about an enzyme, there will be a bond. You don't believe me? You look at all your entire O-level syllabus, A-level syllabus. Every time there's an enzyme, there will be a bond next to it. Even in your enzyme topic, they ask you catalyze hydrolysis or whatever reaction, you need to say, oh, this thing breaks glycosidic bond, hydrogen bond, peptide bond. So enzymes will always be accompanied with a bond. And that means in your answer, if you put the bond name with the enzyme, you get 1 mark. Okay? If there's an enzyme catalyzing that reaction, it's also worth 1 mark. So in short, if you know the reaction has an enzyme name and there's a specific bond, they are worth 1 mark each. Okay. Now, okay, as my DNA polymerase reads and synthesizes the whole daughter strand, let's take a step back. What's the purpose of DNA? Let's ask Jia Ying. What's the purpose of DNA? Inheritance, right? So important, right? Basically, if your DNA is damaged, what do I call it? A mutation, huh? So it's very important that I make sure it's correct. So that is what this step is for. So just tell yourself, oh, as I'm fixing the jigsaw puzzle, okay, if I see a piece that is wrong, what do? You will change it, right? Likewise, when I'm doing DNA replication, something must check that the nucleotides are correct. If it's wrong, it will also fix the problem. Same as you. If you find a wrong piece there, what do? You fix it yourself, right? You won't call your friend, yo, can you come over? Why? Can you help me fix one jigsaw puzzle piece? Your friend will just hang up on you. Right. So leading and lagging strand is going to happen now. And why does this happen? Is because, okay, as you unwind your DNA at the replication fork, okay, because of the directionality, the anti-parallel, you will definitely result in a situation where one part is unwinding, okay, at a disjointed rate. The other part is unwinding in the same direction, okay, as the strand being synthesized. So that's why we get this problem. Yeah, so leading strand, okay, so to answer Xing question just now, how come the gap at my leading strand will be filled up? So you can see now, eventually on the other half of my replication bubble, it will join up. Okay, so when it comes to leading strand, important phrases to write in your answer. It is towards the replication fork and it's continuous. This is the common mistake most students miss out in the answer. Okay, 2 things, towards the replication fork and in a continuous manner. Write that down. Then, likewise, opposite for your lagging strand, it will be discontinuous and away from your application fork. So these are some common mistakes where students reach this part of application when they explain it. They either miss out discontinuous or continuous, or they miss out away from the application fork or towards the application fork. All right. Now, once you reach the lagging strand part, what is synonymous is, oh, lagging strand, there'll be little chunks, little chunks, little chunks. What are those little chunks called? They're called fragments. Why you have to remember the name Okazaki? Because the person who discovered it, Mr. and Mrs. Professor Okazaki, named it after themselves. Rightfully so lah. And because they named it after themselves, you have to remember their name. Okay, Okazaki fragment. So, if I have a lot of fragments or continuously synthesizing like this, so every time it unwinds, I put a new primer, I synthesize that part. Unwinds again, I put another primer, it synthesizes that part. I'll get many, many fragments. Do you think that's good for DNA? If it's very fragmented. Mind you, this is still attached to a template strand. If I remove the template strand, will these different parts be a good thing? It's like you take MRT, if the track not connected, you dare to take? I don't dare lah. That's why I have no faith in the Malaysia highway system. You know, Malaysia got this, you want to build a bullet train to KL. I don't dare. Because if they don't maintain the track, the train very soon from bullet train become an aeroplane. And after that, it's a very short airplane, right? Then when you land, you have a one-way ticket to heaven. So a bit scared. Okay? So now that you know, hey, got parts missing, something must join the parts together. What is this thing? It's called my ligase. Where does the name ligase come from? The chemical process of joining two nucleotides together is called ligation. So ligation, ligase. Okay. Now, we also call that space there called a nick. So you can either say seal the nick, seal the gap, anything is fine. As long as you explain to your teacher, beside my one fragment and another fragment, there is no bond there. What is to form? My phosphodiester bond. So ligase does that. Okay. So once again, enzyme name, sure got bond. Ligase catalyzes phosphodiester bond. Okay. Over here. So if you are a more visual person, what I'm telling you is this. There is literally a space here, and that space means there is no phosphodiester bonds between them. In short, it's like a couple. After the couple argue, they don't hold hand, right? One gap there, lor. Then after they make up ready, they're happy, they hold hand again. That's a phosphodiester bond. Right? In the jigsaw puzzle case, it's after you make your jigsaw puzzle, very nice. I want to frame it up there. But if I frame it up there, and gravity acts on it long enough, it will fall. So I will glue it together. So this ligase is like gluing stuff together, forming a continuous DNA strand. All right. Then eventually you're going to get your double helix back. So at this stage, I'll emphasize, if my single-stranded binding proteins was holding it apart, now after everything is done, the single-stranded binding proteins will just move away. Then it will naturally re-anneal back. Okay, now this re-annealing process, or this part where the separated strands, okay, become back like that, is a natural process. It doesn't need energy. I say again, it doesn't need energy. That's why here you don't see anything about ATP. Okay. Now, in your paper, okay, or in your notes, I have given you a very common answer to the question, describe the process of DNA replication. Okay, make a better term, this is a model essay. You can study it, but whatever I just told you in slides is just step 1 to step 18. Okay? I pray very hard it comes out for your promos. Then you can drop me a text. Mr. Po, thank you. Okay? Okay? Now, moving on to control gene expression. This topic, very complex. Don't worry, it's my job to make it easier for you using K-drama and K-pop. So for this whole entire thing, first part, there is something happening with my promoter region. There's something happening at my DNA. Let's focus at the DNA part first. So the DNA part, I'm interested at my DNA itself and histones. What are histones? Histones are just a protein which DNA wraps around. Okay, hold on. Let me go and get my DNA from somewhere. Oh, here is DNA. The plus of boomer. So I use Scotch tape and this. Okay, so this is my DNA. People at home. Histone, DNA, wrap around, just nice. Next Sunday is my wedding anniversary. I can give to my wife. I'm just kidding, I'm not. Okay. I want another wedding anniversary, so I shall not do that. But essentially, this is what histone do. They wrap around. Okay. Some people are very confused. Sure, you're married. Yeah, of course I have a kid. Of course I have to be married. I cannot have a kid without being married, right? Okay. I'm not so liberal, I'm not so liberal. So now DNA methylation. First thing first, what in the world is that? It's literally adding a methyl to the DNA. Okay. Now, your school may tell you cytosine, some school may teach you guanine. Basically, it's called a CPG site. What is a CPG site? You see here got C, you see here got G in between. So it's the phosphate, phosphodiester bond between my C and my G. So if they give you a question full of DNA, a full DNA nucleotide, okay, a full DNA strand, and there is no C or G, right? Can I methylate it? You cannot. Okay. Now, enzyme involved methyl transferase. Easy lah. I'm adding a methyl to it. So methyl transferase. Okay. Oh, they're taking pictures, so I'm small. I'm just kidding. I'm taking picture, that's small. Okay, so now you must think of DNA methylation as long-term inactivation. So any kind of DNA that is methylated, like just now the question on the lactase thing, right? Means your body don't need it anymore. Okay, so it's kind of like reading a book. When you're done with a book, by right, if you are a good boy, good girl, what do? You keep it in a bookcase or wherever lah. You pack nicely. Okay, if it's something you need very often, do you put it in a bookcase? No, right? You put it on a table for easy access. So DNA methylation is for genes that your body don't need. A question would then be, why do I need to do this? So if you are studying now for promos, and if on your table you still have all your O-level TYS notes, is it very efficient? If where's the notes? You dig, 3 hours later, you still don't find your notes. So same thing as DNA. If I'm a specific cell, okay, for example, I am a stomach cell, I need to synthesize enzymes, okay, like protease. I just need to transcribe the gene for protease. Every other gene, do I need it? No, so I methylate it, okay, because they are going to block me and take up space. If I don't have these, quite scary. So in my eyeball now, does my eyeball need protease? No. Can you imagine if the eyeball protease gene wasn't methylated? One day you are crying tears of enzymes. Okay, so that's why you have methylation. So once you see methylation, it means long-term inactivation. In short, for life. Okay. Then we come to histone. So histone is the orange. Okay, it is a protein. Protein got made out of 8 different subunits. So before I even go to DNA, can I check? Histone is which level of protein? Which level of protein folding is my histone? Just like an orange got 8 wedges. Actually, I don't know. Come. Huh? Tertiary. Okay. Xing says tertiary. What were the rest? Some say quaternary. Anyone say secondary? Okay. Okay. Quaternary. Why? More than one subunit. Okay. So there's more than one subunit. Now, on the topic of proteins, if they give you a question, they ask you to get state the highest level of folding of this protein structure in the figure. It's a hint. They want to trap you. Look at the figure carefully. For a teacher to be able to out-argue themselves when they mark you wrong, they must give you a hint. So check the picture. Sure must have one N terminus or C terminus. Okay? Or they mention somewhere in the preamble that tells you it is one chain. If you see 2 C terminus and 2 N terminus, straight away, there is more than one polypeptide chain. Straight away, quaternary structure. Once again, quaternary got R after the QUA. How you remember? Okay, okay. Now, histones are positively charged. How to remember this? What is the charge on DNA? Why negative? Why negative? My GP brothers and sisters. I hear something very loud. Why negative? Thank you. Call the phosphate group. Okay, so the phosphate diester bonds are there. Phosphate is negatively charged. Your whole DNA is negative. Now I know why my friend very negative. Yeah, that's right. The DNA very strong. Okay, now joking. So if DNA is negative, histone must stick to it. They must be opposite attract. So it must be very positive. So in life, please aim to be a histone, be more positive. Okay? So they come together. Now, question, how to remember the acetylation or deacetylation is the one that make it more or less accessible? Very, very simple. Use English. Acetyl sounds like excess. If I put the acetyl, I'm putting excess. So if I have more acetyl group added, my transcription factors can access it better. If I can access it better, is it very tightly wrapped up or very loose? Very loose. So you just use the English word excess. Once you see excess there, whoa, okay? So if it's de-excess, means no excess, it's more tightly wrapped up. And with that, this should wrap it up and make it very easy for you. So just use the word excess to help you. But please don't spell acetylation as ACCES double C. Don't spell wrongly. Okay. Now, so pictorially, once I have my acetyl groups, I have excess. Excess means what? It becomes less tightly wrapped up. Less tightly wrapped up means, it should be less positive charge. That makes sense? Less positive charge. My DNA negatively charged, they won't bind as much. So less tightly bound. There is more space now like this. When there's more space now, what happens? Your transcription factors, your RNA polymerase can enter. Okay? Now, both processes will have an enzyme involved. Adding acetylase is called acetyltransferase. Removing it is called deacetylase. So just take note of which enzyme to mention. Yeah? So I learned about DNA methylation. I learned about acetylation on my histones. Both work together at the same time. So logically, if something is methylated, means long-term silence. Do I want access to it? No, right? So it should be deacetylated. Just so easy. Yes, that easy. Okay. Now, next question. How to remember exam? I really forget whether is it histones get methylated or histones get acetylated. Very easy. K-pop era, right? So who is this? No, these are people who are doing national service in Korea. Okay, I don't know who but to me that's very strange. I just heard, okay, so this fella in the middle is serving NS now. But this fella's song in Singapore is #1 on the 987 charts. I don't know how that works. This is Jimin, by the way, okay? Why I know? I'm not a BTS fan, but I have a colleague who is. And she's super crazy about them. So I know it's Jimin. The rest, I thought were the same, because the face was similar. Okay? But their songs are nice. Now, how to remember it? Okay. BTS has dope moves and they always hype. So dope moves hype always. Okay? This is how my analogy to you. Dope moves. Do you see the song Butter? You see how they dance in Butter? Oh, the moves really damn dope. And they're always very hype. So dope moves hype always. Okay, that's how you know. Actually, you just remember dope moves. Then you know show D and M together. So D for DNA, moves for methylation, hype for histone, A for acetylation. Okay? Dope moves. BTS has dope moves. Don't worry, they will appear later again. That's part of my lecture. Actually, they're not coming here. I'm just saying they appear on the screen. Okay? If they come here, wow, Mr. Hua. Sign up for the Okay, now we move to the control part. So in the control part over here, this is a DNA strand. So we are now looking into the different regions. We have the enhancer region or silencer region, promoter region. We have my coding region. We have another enhancer and silencer. So the terms you must be familiar with is this. If it is involved in controlling, aka it doesn't code for a protein, it's called a control element. So note this down. If it doesn't code for a protein, it is called a controlling element. What codes for your protein? Here. Only your exons, okay? That gene region is your protein part. Everything else is called a control element. And because they don't code for a protein, we call them non-coding sequences. Oh, yeah, makes sense, right? Yeah. Now, so different types of transcription factors will bind to different control elements. Firstly, at the promoter. So your promoter is where everything happens. So there's different levels of transcription. There's very fast, there's very low. If I want very low level of transcription, the bare minimum, right, Okay, bare minimum is where the word bare comes from. Basal stands for bare minimum. So I have basal transcription factors. Okay. So where they bind to? They bind to the promoter. Because the promoter is where everything begins. Next, I have this thing called activator. Makes sense, right? Activator should bind to an enhancer. Then the other one is repressor and silencer. But students have this headache. Sure, how do I remember which one is the DNA, which one is the protein? Very simple. Activator and repressor. What is the second last alphabet? O, right? DNA got letter O. Protein got alphabet O. So those that have the word OR, activator, repressor, alphabet O there is the protein. The one without the O is DNA. So if someone asks me about enhancer and silencer, I am looking at DNA. Good, you copy down. That's liquid gold. Okay, good. Okay, so enhancer and silencer ER. No O, not a protein. There must be DNA. Activator for a O should be protein. Okay. Now, so your transcription factors are like this. You have your basal level, you have your specific level. Okay. End of the day, they bind together to make it fast. Basal is what I need for normal speed of transcription. Everything else here, the specific one, makes it faster. So once you see in the terms, oh yeah, it's quite easy, quite straightforward. Then this part keeps you confused. My activator protein can bind to the enhancer sequence. So if it can bind to the DNA, it has a DNA binding domain. If it can bind to a protein, it has a protein binding domain. How does this work? It just, once again, shape complementary to shape. So when you see a question on DNA binding domain, it means the shape of this binding site is complementary to the shape of the DNA. DNA got shape, man? Yes. Your A, T, C, G are different nucleotides, right? If I put them side by side, I get a specific shape. So if I get a different sequence, I get a different shape. That's all. This is just complementary binding. That's all. Okay. Okay, I'm going to skip this. All right. Okay, then once again, now back to the part here. Enhancers increase transcription, quite straightforward. Repressors, okay. Now, so how does the enhancer increase transcription? First part, it has to make your gene more accessible. Some link to your acetylation. So more accessible, it should be acetyl transferase. Okay, then all of this, you know. Okay, this part over here. So one thing to know is this: once you have formed this part over here, your basal transcription factors band here. I have my specific transcription factors. I have my basal transcription factors. Your DNA will bend, okay, and make it more stable. What is being more stable? Your TIC. So, in short, it's like, let me see this on the screen. Okay, this thing here, data box. Okay, if a basal transcription factor, I put an orange here, and I move the orange will fall off. Okay, a bit hard for you to see. Let me see like that. I put the orange here, and I took this. Sorry, you won't say that, right? Then what are other better transcription factors? Your proximal transcription factor. I put it here. Like this. Okay. Then specific transcription factor. I put one more there. It's like, what is this old man doing? Wait for it. After I put this here, then your enhancer, right? Activated protein, bind to it. Put together here, what's going to happen now? This thing is so stable, I can throw around. The orange is not flying. Do you see? And if it's so stable now, what's going to happen? This RNA polymerase will shoot out so fast and transcribe very quickly. This is the part where you clap. Okay, so you get it now? All these proteins just help make it very, very, very stable. Go back home. How was the lesson now? Oh, the teacher played orange. All right, now, so your analogy is like this. You can think of it, okay, your activators, enhancers, everything, all as you at home. Now, if I want to watch Netflix and very comfortable, what do I want? Wow, my favorite bubble tea, Chicha, premium stuff. Okay. Then I got my favorite snacks, Cheetos. So then after that, I have a soft toy to hug. Okay. Then far away, maybe I burn some incense for the smell. Then got the mood to watch show, right? Then if all of these, okay, are like activators. They come together to make the perfect environment for your RNA polymerase to be stable. So when I have all of this at home, what's going to happen? I'm going to be a happy guy. I watch TV. Okay. Then reverse. What do repressors do? Very simple. Now I need to go toilet. I don't want people to sit in my nice comfortable chair, drink my chicha, and hug my soft toy. What do? I try to make it as uncomfortable now. So what's the easiest and fastest way to make this uncomfortable? I just put tongue side over here. I put it there, what happens? No one's going to sit there, right? Likewise, this is what a repressor does. When a repressor binds to the same spot over here, so if the repressor now binds here, can this protein bind here? Cannot. Okay, so repressors are just simply irritating people who will just sit at places they shouldn't sit. Okay, so once I put the repressor here, RNA polymerase cannot bind. My activator proteins cannot bind. Okay, so simple analogy lah. Okay, so when that happens, that's why you have a decrease in your transcription rate. And as you can see in the picture, this repressor very annoying. Binding here, and it overlaps with the activator binding site. Okay. All right. Now, I will give you a free tip, but I'll skip the rest. This one, another headache you'll have. So how to remember, you know what's this? Your DNA fold into your nucleosome fold into solenoid fold into chromosome, right? You cannot remember the name? Okay, no worries. For those who need to know, the answer was just now on TV. During the break, what was I playing? What song was that? Cookie, right? Who was singing it? Besides singing, what were they doing? Okay, so order Dancing New Jeans singing Cookie. It's like, what the lame. Oh my God, remember this down. Okay, so Dancing Cookie singing New Jeans. Sorry, Dancing New Jeans singing Cookie. Oh my God, it's been a long night. Okay, now the thing is lame, trust me, when respiration comes, it will tickle you so much, you will laugh. Okay, so now respiration, I'll go straight to the part which is very painful. Glycolysis is very painful for you all because you all don't really approach it correctly. Now, I see glycolysis like making lemonade. Why? A lemon is like an oval shape. Okay, oval shape to me looks like a 6-carbon ring. So it's kind of like your glucose. Now, when I want to make lemonade, what do? I must first buy lemon, right? Lemon costs $2. Wait, at what time? 8 o'clock, right? Yeah, why am I rushing? Go back. I thought I end at 7:30. Then I see you're so stressed. Hey, okay, come back here. Anyway, my emergency plan is when I finish, right? You ask me a topic on the spot, I teach you. So, sorry, let's go to this. Here, as you can see over here, it interferes with the egg. So it will bind and block there. So one way it binds and blocks the blocking site, or it binds to the activator directly and prevents it from moving. Okay? Lucky you'll give a panic face. Then I'm like, why are you panicking? Oh yeah, I got so much time. Okay, next, just as how, okay, this thing must bend over here and cap. It can also just block, okay, other proteins here at my TIC. In case you don't know, this whole thing is called a TIC, Transcription Initiation Complex. English very simple. It initiates transcription. Okay. Now it can even block the bending of the DNA. And why do I make this thing? Because whenever I talk to kids about bending DNA, they have no idea what bending DNA is. They cannot imagine. So this is literally bending. So if the DNA cannot bend, it also cannot stabilize. It prevents the TIC performing. What can prevent it from bending? Repressor bind here, prevent it from bending. Also possible. Okay. Now that is at the DNA level. Now in terms of packing, if they want to really slow down transcription, what can they do? Prevent excess. So they can recruit the deacetylase to remove the acetyl groups so that now it is really not accessible. This is the best way to stop transcription. Okay, this is the best way. Once it's so tightly packed and my transcription factors cannot access it, you will get very little transcription. Okay? Raise your hands if you felt that when he walked past halfway, he'll go into some dance move. Yeah. Okay, now modification part. Okay, over here, this portion kids find it very complicated. Here is glycosylation, add this, add that, ubiquitination. Actually, you learn this as the first topic in J1. Where? Remember, her face like, where God? Let me prove you right. Prove you wrong. So remember in cell orchard structure, when you finally had to learn about the Golgi apparatus, what do they say the Golgi apparatus do? Oh, the Golgi apparatus sorts, modifies, and packs. What is the modify? Well, the Golgi apparatus will chemically modify. What is the chemical modification? Ta-da! This. Okay? So what does it add? Simple. It adds a sugar group. So glycosylation, adding sugar group to get glycoproteins. Don't worry, by the end of, towards the end of the A-levels, after you learn genetics of viruses, you'll be very familiar with what's a glycoprotein. Okay? Phosphorylation is just adding a phosphate group. Ubiquitination, besides being very hard to spell, okay, what is this? It's simply, this is the trash bin in the cell. Now at home, as if you have your own room, okay, how do your parents know what they can throw away? Only things in the rubbish bin, right? If they suddenly go to your table, your wardrobe, and throw away everything, you will scream and go crazy, right? So isn't that what the dustbin is for? So same thing in the cell. Now the cell has no eyes. The cell only works on what? Shape, complementary to shape. So if the cell needs to know what is useless and can be thrown away, they will tag it with this thing called ubiquitin. That's so nasty. Okay? So ubiquitin is added to a protein to tell the cell, hey, can the rubbish collectors can remove this protein? I don't need it anymore. Yeah. Okay. The free tip done already. Yeah, one more time. Now I can show the video. The pointer. Okay. So what I need you to look at here is this. It's going to form the TIC, the transcription initiation complex. It will form very quickly, and the moment once the thing stops dancing, it will not just, okay. RNA polymerase will shoot out very quickly and they get transcription. Okay, so here you go. So that's our basal transcription factors.
See is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left.
Okay, that's a second transcription factor.
The assembled factors trigger the first phase of the process.
Another one, a third one, because they're all homo.
That will be needed to make the protein.
Okay. Now that is the DNA gonna bend, huh?
Everything is ready to roll. 3, 2, 1, go.
You want to see that? Jolene missed it because she's looking at her phone. Okay, so show you one more time.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein.
DNA is bending. Now this cup is going to come.
Everything is ready to roll. 3, 2, 1, go.
I'm trying very hard to understand what you're laughing about. Just to be clear, is the video not me, right? Suddenly I started checking my pocket, checking my hair, or checking him, laughing at him, is it? Right. No, just kidding. I don't know what's that. Okay, now let's go to respiration. So back to the lemons. So like I said, the story lemons. Lemon reminds me of what shape is this? Huh? Hexagon, right? So it's like a glucose. So glucose is a 6-carbon ring I thought I took him. Okay, 6 carbon ring. So it reminds me of a lemon shape. Now, when making lemons, what's the first thing you do? You must buy lemons. So when I buy lemons, I spend $2. Next, when I make my lemons, what must I do? I must cut my lemon into 2 pieces. So if my lemon is 1 whole piece, okay, I'll cut the orange now. Just kidding, I won't. Okay, very messy. But in short, I cut my lemon into 2 equal pieces. And if my lemon was a 6 carbon ring, when I divide a 6 carbon ring equally, you get 3 carbon, 3 carbon. Okay? Then now I got 2 3 carbon. I can sell. If I buy for $2, I obviously must make profit, right? So I will sell it for more. I sell for $4. So after selling for $4, what's my profit? $2. Very easy. Okay? At the same time, lemonade. What does it taste like? Sour, right? It's acidic, right? In short, this is exactly like glycolysis. Why? Because glycolysis, you start at 6 carbon. You're going to cut it and break it into 2 carbon. Eventually, you get a pyruvate, which is pyruvate acid, which is acid. Okay? Now, if you're laughing at this, it gets even better. You only learn, is it? Okay, you all learn now for me. Okay? Sorry, JP brothers and sisters. Okay? All right. So, now, starting part, you need to invest energy. Why? Basically, every time I add ATP and ADP here, this molecule becomes very unstable. Simple analogy, yeah? You all know what is glucose? What is glucose? Table sugar, right? Now, your sugar at home, is it very stable or very reactive? Stable, right? How stable is it? I'm very certain when you visit your grandmother or grandfather's house and you want to drink coffee, they ask you, darling, you want sugar? You say yes. They'll bring back out this antique sugar jar. This antique sugar jar, what they do? Oh, left a bit, I fill up. I fill up. So it never ever reaches the bottom. So can you guess how old the sugar is at the bottom of the jar? Probably older than your parents. Because why? Doesn't spoil. That's how stable glucose is. So if something is so stable, how can I make it unreactive? It's basically just like what the Channel 8 drama, the Vasantham drama, or the Chinese drama do in Singapore. If there's a very stable couple, how do we break them up? We spread rumors. We create excitement. We put energy into the relationship. So same thing over here. That's what ATP does. Because glucose is so stable, the body, the cell has to make it unstable first by putting ATP there. Hence, I have 2 ATP molecules there. From glucose, I add a phosphate, glucose 6-phosphate. From glucose 6-phosphate, I add another ATP and I change it. It becomes fructose 1,6-biphosphate. If you're thinking, sure, this is such a pain to remember, let me share with you some happy information. You're very lucky, Eddie. During my time, I had to memorize all of this and all the enzymes, all the names. You all don't need any. So that's good. Okay, right. So long story short, they just do all of this to make the glucose unstable enough that it can break into these 2 things. Why is it called triose phosphate? Because it's 3 carbon and it has a phosphate. For your own sake, also know that triose phosphate is called glyceraldehyde 3-phosphate. You can write triose phosphate in your exams, it's fine. But some questions don't write it as triose phosphate. They will write glyceraldehyde 3-phosphate. So just know both. Okay. And after the whole long process, there's substrate level phosphorylation, okay, as well as producing your NADH to eventually still give you 3 carbon pyruvate. So you start with 6 carbon, you end up with 2 times 3 carbon. I start with 1 lemon. I have 2 half lemons. Don't worry, I know it's an orange. Okay, so, right. So I invested 2 energy worth of ATP. I need to get back, okay, some profits. My total returns is 4 ATP form in total. 2 from each particular triose phosphate. So if I have 2 formed, but I invested 2, what is my net profit? 2 ATP. Okay, same thing. I buy for $2, okay, end up I earn $4, my net profit $2. So now comes to this. So I know the process. I don't know how to remember the process. Okay. So how to remember the process? BTS is here to help you. So who is this fella? His name is who? V. According to online, it's called Tay also lah. Okay. So, yeah. Okay, what was again? Bro, you must understand, the girls who are posting on X, formerly known as Twitter, okay, or IG, I stand it. Very long. I stand D easier. Okay, it saves you so much time. Now, so fella can sing very well. Songs are very good. I forget whether he's the one who likes IU or the other one. One of them likes IU. Yeah, but yeah, correct. Wow, who's the you? Is it? Yeah. So I know the one that likes IU didn't get to sing with IU because fella in NS. This fella sang with IU recently. Then he went to NS. Not my choice. Later I tell you why. Okay. So now what is this prize at the bottom here? That's a Golden Globe Award. In short, you sing very nice music, people like, they're happy, they vote, they give you a prize. It's the top prize for music. These two together will help you remember the order. So basically, it is Golden Globe for fantastic taste performance. That was a tad loud. That was a tad loud, sorry. Okay. That's not a BTS song, for those who don't know. Okay. Right, so Golden Globe for fantastic taste performance. Golden, G for glucose, Globe for glucose 6-phosphate, 4 for fructose, fantastic for fructose, phosphate, okay, and T for trials phosphate and pyruvate. Running Cher, is it okay to skip so much? Yes, it is in your revised syllabus. You don't have to know what's in between. Okay, okay. What you have to know is this, the enzyme. So this part go, no, it's not in your syllabus. You don't need to know the enzymes. So just know this. As per all analogies, it's very good if you can remember it, but please don't write this in the exam. Don't write Golden Globe for fantastic day. Your teacher will, okay, please be able to link it to the actual thing. Okay. Now, next part. Okay, I'll bring back to this slide. Once again, remind you, okay, the only thing you have to know about me today is Suzy and IU. Why? Because the moment has come. I've been so excited the whole day to teach this part. Okay. Now, Krebs cycle, the bane of your life. Why? Krebs cycle, kids find it very painful because a lot of long names hard to remember. And I have no idea what to remember. So, to my dear JP friends, after today, I guarantee you, you will remember this, even though your teacher haven't teach you. I'm quite confident. I've tested it among many people. It is the greatest level of nonsense I can provide you, but I'm quite certain you remember. So, how does the Krebs cycle work? It starts with a citrate. Now, a lot of people focus on the alphabet. Wrong. You should focus on the carbon number. So, what enters this whole reaction? This thing called acetyl-CoA. It has 2 carbon. Okay, so I skip the link reaction for those. So, 2 carbon plus 4 carbon. End of the day, remember the lemon. The whole entire respiration is just about the lemon. Lemon got 6 carbon. So every process here will go back to a maximum of 6 carbon. Okay, so 2 carbon plus 4 gives me 6. After citrate, okay, I get alpha ketoglutarate. When this thing happens, carbon number 6 becomes carbon number 5. Right? So clearly, I've lost a carbon. In respiration, when you lose a carbon, you are losing it via carbon dioxide. So the reaction is called oxidative decarboxylation. Because we are not chemistry, we don't really care about the oxidative part. We care more about the decarboxylative part. Okay? Now at the same time, because citrates oxidize, something has to be reduced. Because that is how karma works in the world, right? So what is being reduced here? Your NADH. So this whole reaction happens here to produce NADH. Reduce NAD. Now comes to alpha ketoglutarate. Okay, 5 carbon. 5 carbon AD, same thing, undergoes a second round, I get succinyl-CoA. I have 4 carbon now. It will stop here. It will not go any lesser. Because what enters is acetyl-CoA, which is 2 carbon. So 4 plus 2 give me 6. 6 minus 2. This succinyl-CoA, which is 4 carbon, will stay 4 carbon all the way. Right? Then succinyl-CoA undergoes substrate level phosphorylation to form succinate, malate, so on and so forth. This one you all know. Sure, teach me how to make this easier to remember. First thing first, I will teach you how to remember the carbon number. Okay? Now you have to indulge me. How do you pronounce this word? No, the whole thing. Okay? Most people will go alpha ketoglutarate. This is how I pronounce it. Alpha, keto, glutarate. I just realized I cannot see. One more time. Alpha, keto, glutarate. Now, I'm wondering, why is this weird guy in a man bun doing that with his hands? Alpha, keto, glutarate, 5 carbon. Oh my god, just so lame. Yeah, but you remember. So there's a chant to it. The chant goes like this. Okay. Alpha, keto, glutarate. Please don't do that in exam. Okay, but generally, the tempo, alpha, keto, glutarate, 5 carbon. So from 6 become 5. Then now, oh, what nonsense is he going to teach me for succinyl-CoA? Same thing. Now change hand. Okay. Succinyl-CoA. Succinyl-CoA. Your generation learned the word sus, right? Now bring it to practice. Succinyl-CoA. Now to really make you benefit from this, to the people at home, I want you to indulge me. Can you at home, 10 times on your own, right now, do alpha, keto, glutarate. Go. Do it. You do it, you remember this really silly thing, and in exams you won't forget. After you do it 10 times, you go succinyl-CoA. 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 You think, this is very lame. Okay, I've taught this to a class. They thought it was very lame. I forced them to learn it. In that class, 5 are doctors now. And the exam, they say, hey, I already do the thing in the exam. How I know they do the exam for A-level? Because my colleague walking down that row, hey, Glendon, why your class students so big on? The exam halfway day. What are you doing? Secret. Okay, it works. So you can say it's lame, but lame stuff work, you remember it. So, okay, he's taught me one lame way to remember carbon number. Right? Now, how to remember when, what alphabet comes here, what energy, this is the time to teach you now. So, firstly, how to remember this? Citrate, okay, starts with come. Okay, next, come and kiss. Alpha, keto, glutamate. Okay, come and kiss who? Remember just now my story? Sweet Susie. Okay, come and kiss Sweet Susie. What's M and O going to be? If I get to kiss Susie in my life, do you think I'll kiss her once? No, I'll kiss her more often. So now here the thing is this. Mr. Pua, what the? But never mind. With this, you can remember what is the order of things. Come and kiss Sweet Susie more often. Sure. How does this, you all don't take picture first. It gets better. How to remember where all the energy come from? Simple. When I want to do anything in this story, if I need energy, there's NADH. Put to you. Now, if I want to come towards Susie to kiss her, I must walk, right? Come and kiss. So between come and kiss, I must move towards her. Energy, NADH. Okay? Now, when I kiss someone, need energy or not? Okay, I don't know if you all got experience. I got a lot because I'm married. When you kiss someone, sure need energy one. So, NADH. Now, Sweet Susie, no kissing involved. But why got energy? How to remember this energy here is ATP? Very simple. You all know which band Susie is from? What? Susie is from Miss A. Oh my God, just stars. So, Miss A, ATP. Sweet Susie is from the band, original girl group called Miss A, ATP. Okay? Then, how to make FADH? I'll change the story. Come and kiss Sweet Susie's friend more often. You all know who is Susie's very good friend? I'm not bluffing you. You can Google. Susie's good friend is none other than IU. Okay? Come and kiss Susie's sweet friend more often. Okay? Alright. In all honesty, I like her more. Okay? I think her singing is very good. Susie, good actor. She has become a very good actress now. Every time her shows come up, I binge watch with my wife. What's her latest one? Okay, really good. Okay? Now, the nonsense doesn't stop there. So now you know, alpha, keto, glutarate, succinyl-CoA. Okay, your friends are missing out, they're not here. And come and kiss Sweet Susie's friend more often. Okay, lovely. So, Susie's friend, the F here is for FADH. Now, when I kiss someone more often, obviously I need more energy, NADH. Okay. Oh my God, Jordan Lim. But yes, you remember from now on. Hey, please don't write the kissing part in the exam. Please write the proper thing. Okay. Now, next part. Because the kids cannot remember, at the end of the day, they always ask me how many NADH formula, how many FADH form, how many ATP form. I cannot, it's too much. Let's go check a bit. When do you publicly kiss someone and everyone claps? Only one scenario in life. When is the one moment where you publicly kiss someone at a wedding, right? So what is the wedding mean with this? Very simple. What is the song you all sing at a wedding? Yeah, for animation. Okay. Now, what song do you play at a wedding? The very typical one is no money. What's the typical wedding theme at church? Hey, no lah, church will be 1,000 years lah. Now go check. Then wedding banquet. Okay. Okay, you know the song? Dun, dun. Okay, now I know what this will play at their weddings. So you know the typical church, the organ, ten, 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 ten. So how is that link? Very simple. At the end of the day, you form 10 NADH because 10, 10, 10, 10, 10, 10, 10, 10. Then how to remember how many ATP form in total? Remember, marriage is forever. So 4 ATP is 4. And then how to remember FADH2? Quite easy lah. Marriage is a union between 2 people. What the hell, Cher? You sure? Yeah, see, my nonsense top tier one, don't worry, but you have to remember. Okay? So in the exam, how many DH? 10, 10, 10, 10. Then you have 4 ATP form at the end of respiration, 2 at glycolysis, 2 at Krebs cycle. Why 2 at Krebs cycle? Because of Susie. Okay? And my marriage is not to Susie. Okay, Ken? And it's between 2 people. All right. Okay, ready? Now, jokes aside, let's move to the more less interesting one. So oxidative phosphorylation. In short, I need to use my NADH and FADH2 to make more ATP. So here, the main thing is this: FADH2 and NADH are electron donators. They will donate electrons to this whole entire ETC. What are they doing? It is this. First thing first, let me orientate you to the fact that there are 3 different proteins here. Then each protein got an arrow going up. Each arrow going up is like H plus proton being pumped up. Okay, so where does NADH start the whole process? NADH starts here. So you go through 1, 2, 3. That's why it will pump a total of, look at the electron. As the electron passes through, okay, it will pump up H plus. FADH2 starts at the second one only. Okay, there are implications for this. At the end of the day, who's the final acceptor? Your oxygen. That is why we must breathe. Okay, that is why we must breathe and take oxygen for this step. Not enough oxygen from very strenuous activities such as more kissing. That's why you have to go towards aerobic respiration. Okay, so you have to know O2 is the final electron acceptor. Now, over here, as the E flows through, when my NADH electron flows through the first protein, a proton is pumped up. Sorry. When it goes through the second one, you pump a second one. It goes through the third one, it pumps a third one. So in total, how many H plus would my NADH have pumped up? 3. And with 3 H plus, each H plus will be used later to form ATP. That's why your NADH forms 3 ATP. Okay? So now, if they ask you a question, how is the steep proton gradient maintained? This is not a respiration question. This is a membrane question. Why? It's a membrane, the hydrophobic boundary cannot pass. Yes, it's here. So for the membrane here, okay, H plus pumped up there, H plus cannot go through, cannot cross the hydrophobic core of the bilayer. So it's not something new. It's using what you learn from membrane for this part. Okay? So next, H plus will diffuse down your ATP synthase, and the energy used, okay, will release and is coupled to ATP synthesis. If a question asks you about chemiosmosis, this part here, 6 and 7, is chemiosmosis. So chemiosmosis specifically is just this part. It is a 2-mark question. If they ask you about the whole ETC, electron transport chain, plus chemiosmosis, is usually about 4 to 5 marks. So again, if you are asked to describe the whole electron transport chain, it's about 4 to 5 marks, including chemiosmosis. Chemiosmosis is just 2 marks. What is chemiosmosis? Chemical. Osmosis, something moving. So H plus is moving down the ATP synthase. Energy release is used to synthesize ATP. This thing is called chemiosmosis. Okay? Now the energy level die already. Why got no more wedding? Nothing. Okay. Not be too happy. Okay. All right. So it's just like that. Hey, too fast. Okay. Now at this stage, yeah, I'm going to something. So NADH will go through 1, 2, 3. FADH2 will go through only 2. That is why, if I go back to this slide, all the way here, that is why your NADH will give you 3 ATP molecules because it pumps up 3H+. Your FADH only pumps up 2H+, it gives you 2 times 2 gives me 4. So after I add up everything in respiration from glycolysis, I have net 2. Krebs cycle, I have 2. Okay, plus 34, I get 38. Okay. Okay, very good question. So why? It is because of this page here. So do you see where NADH releases the electron? At the first one. So you see, and I release here, 1H plus pumped up. This electron continues here. A second H plus pumped up. Continues over here. A third H plus pumped up. The H plus is from the matrix. Okay. So very simple to equate. When 1H plus goes through the ATP synthase once, you produce 1 ATP. So if FADH releases electrons here and it passes through 2 proteins and it pumps up 2H plus, it produces 2 ATP. That's all. Okay. Okay. All right. So that is all I have for this part over here. I've saved some time to add other areas. Yeah, before I go to this. Can I check any one of your being tested genetics of viruses for your promos? Whoa, okay, let me help my friends from Jurong Pioneer. Okay, then I can continue. Okay, the amazingness. Do. Yeah. You're gonna be. Let me put here. Wrong one, sorry. Okay, I'm for the, who got viruses? Okay, so I'll just go through the part which is very painful for you. The phage not too is difficult. Now for phages, the thing you all struggle with is genetic material. Now the genetic material is DNA and there's RNA. Okay. Now DNA is only for phages. Let me make it bigger. Phages. So you have a lambda or not? DNA is always double-stranded. Okay. DNA is double-stranded. So both your phages, if it comes out, it's just simply lytic lysogenic, is double-stranded. The issue you have with RNA viruses is influenza. Correct? And HIV. How to remember whether the RNA single-stranded 8 number? Okay, the answer is actually here already. Sure, what do you mean? Let me write. Now, how to remember that my influenza firstly has negative RNA? It is staring at you in the face. Here, negative sign. Then the individual here, this tells me is single-stranded. Okay? Then, how to remember got 8 pieces of RNA? The answer is there. Can you count how many alphabets are left? 1, 2, 3, 4, 5, 6, 7. Oh my God. 8. So I addressed 2 of the painful points already. Next one, the process. Wow. How to enter, how to leave. Okay. So when it enters, okay, that one will enter through endocytosis. So how to remember? Endocytosis. It's there for you already. Okay? Then it will bud off. So HIV, how to remember the genome? That one, also very easy. Now, if ever someone talks to you in life about HIV, what is the thing you are most afraid of? You heard the word HIV. HIV something. Positive, right? HIV positive. So the RNA is positive RNA. Thank you so much for it. But you thank you so much for it. So it is positive RNA. All right. Then how to remember? How to remember this? 2 copies. Very simple. 1 copy, 2 copy. The alphabet H. Okay. Well, if I knew so people needed viruses, I would have taught viruses. Okay. Now, so influenza single-stranded, use the I to help you. Negative sign, negative sign. Use the middle part of the I to tell you is single. So it only has single RNA. Means the RNA is not double-stranded. And it has 8 pieces. 1, 2, 3, 4, 5, 6, 7, 8. By spelling out the remainder of the alphabets. HIV is positive strand. Because HIV positive, you are afraid of it. Now, how to remember that? Okay, let me just do this. September EOI event. Dollar is there. Thanks for holding on for so long. What time now? Let me show you. Okay, now this part. Okay, for the virus people, how to remember that starting it involves your hemagglutinin and neuraminidase. Can I get Satya to come in front? Because I can only do this with you. Because you're the only guy. Okay. So what they find difficult to remember is basically how to remember is hemagglutinin and how to remember is salicylic acid receptor. No, bro, I won't hear you. Okay, so when 2 guys never meet a long time, what do? How do guys meet, Satya? Shake hand, right? Hand. HA. Pass. Then in typical Singapore fashion, if I very long never see my brother, the first time I see him, you know, I'll say, Siala bro, long time no see. Siala bro, salicylic acid receptor. What the? Okay, that's how you remember. Okay, thank you so much. Give me a round of applause. Okay? No, my hand clean. So, hemagglutinin, she missed the funny part. It's okay. Okay, later I'll show you. Okay? So, basically, HA for hand, for ha. Okay? When 2 brothers long time never meet, they'll shake hands. And the first thing, a typical Singaporean guy like me will say, Siala bro, long time no see. Siala for salicylic acid receptor. Hence for hemagglutinin. Now, okay, how does it enter? Endocytosis. And what I said is in the name, influenza. HIV got no EN. So how does HIV enter? It fuses. That's all. Okay, then over here. Right. Now, when it comes to HIV, how to remember the proteins on top? That's why I'm not bluffing you, but I was kind of born to teach bio and teach this topic. So the things that students struggle with my dear HIV, is this. The protein receptor. Where is it? Okay. Now, Where's my HIV? Okay, here. Now, on HIV, what is the protein that we are interested in? The glycoprotein is called, okay, GP120. My name is Glendon. My surname is Phua. My birthday is on 20th January. So GP120. Legit, not bluffing you. You can Google me. My birthday is there. Okay? So when I tell my kids that, oh my God, usually in school, by the time I teach this, it's just nice near my birthday. So you know what happens when I tell them my birthday is coming? Of course, I get present. You know what's the present? And then I bring my homework. Okay, so GP120, this really works because they like me. They also my kids like GP. This kid now is in FASS. She's not doing bio, but every year she wishes me happy birthday because of GP120. Okay, can? Then GP41, how to remember this? That's my IQ. That's kidding. That's not my IQ. GP41, unfortunately, I have no way to help you remember it in a funny manner. But GP120 is me. I will help you enter the school you want in university. Likewise, GP120 helps the HIV virus bind to and attach first. Then 41 is the part where it fuses and everything. Okay. Now, in lieu of time, I would love to chit chat here until 12 o'clock. Okay, but I need to move on to the next part of the sharing. I enjoyed having you all here today. It was very, very fun. I was very excited to teach this. I love teaching bio, a bit of a nerd. But if you enjoyed the session as much as me, before you head to the nearest McDonald's to eat your McSpicy. Okay, right. Could I please invite you to dying and nearly know what's going to happen next? Can I invite you to please fill out my feedback form to show me? What? Yeah, it's all here already. Yeah, it's all ready. So once you are done with the survey, just come to me, show to me, and I will show about the feedback. Okay. Now at the same time, okay, at the same time, I have laminated periodic table. Oh, overmark is the gift that keeps giving. Yeah, that's right. Okay. So people online, it will help me a lot if you can fill up the survey as well. Whoever is left behind, all 17 of you. But yeah, you won't get a full scale pet, but I really appreciate if you give me feedback on my teaching. Okay. Thank you so much. Take your time. That's fast.
I put that for everything.
Oh, thank you so much. Thank you so much. You can just show me that. Extra or what? Are you regular student?
So can I use like I got 3 in right? So all 3 I did already.
Sure.
Okay. Damn.
Thank you so much. I hope you enjoyed the lesson. Sorry, I just use balance. First, can I write down in color of the baby? Wait. These kind of other letters like math and EP.
Yeah, I did.
Then you do like 3 figures for all 3 and then yeah, her name is no, come for another lesson maybe for you lah.
Online people don't get it.
Just stop the final for you. Betul tak? What? I didn't do the survey, sorry. Okay. Right, yeah. Those are that. If not, one last slide. Okay, for those of you, if you enjoyed the lesson and need help, we are doing intensive booster classes. If you are interested to find out about the booster classes, you can scan this. Okay, so need for people who are interested, right? Okay. Okay. See, for the people online, if you're interested in joining for booster classes, okay, you can just scan this QR code and get more information. It's not. So, thank you so much, everyone. I wish you all the best for promos. Okay, I don't see you anymore. What's happening, Gabriela? Nothing. I trust you. In person,
Yeah.
uh,
There's like a whole row of like table.
let me ask you, is there math slot for Roman math? Any day,
Any day.
any day, Roman. Okay, not most who are done. Thank you so much. I appreciate it. Okay, please get home safely. Okay, those that are interested, gonna ask me, can ask me. I'll stay around for a bit. Yeah. Huh? If you that up, that come. They'll try and find you a chair lah. Hey, you can't a chair, right? Thank you so much. You can make your list. Bye bye. Get on safely. First, go and eat spicy and McDonald's. Bye from the west. Thank you so much. Okay. See you again next time. Yeah, let me just